Morning.
Howdy all. Glad to see that at least some of you survived yet another exam day. So onward and upward to our fourth and final portion of this class. We are rapidly approaching the end, which is so crazy how quickly this all goes. But we are on to our last major topic, which is the nervous system. Today, we will talk about nervous tissue. We've talked about muscle tissue, everything you ever wanted to know about more, and muscle tissue, epithelial tissue, connective tissue. That only leaves us one left, nervous, which apparently I can't spell. So we will talk about nervous tissue. Then we will see how far we get today, talk some brain anatomy and histology. We'll see where we are at the end of lecture and go from there. We're not going to get majorly into that handout that you have, but I will tell you that that brain anatomy handout that you have will likely be about 50% of your lab exam. So we will go through it like we've gone through some of our other stuff, but we will also go through it quickly. And then a bit more histology on this test than there was on the last one. The last two, really. Fair amount of homework. We haven't had a Labster in a while, so your first one for that, I guess your last one, I think, for that. Yep, last Labster is Monday, due Monday, so that'll keep you busy this weekend. Remember, it's been a while. You must complete 100% of it and get at least 80% correct. Remember, if you don't get 80% correct, you can turn around and do it again and improve your grade. All right, because if you don't get 80%, you get a score of like 15 points. Or whatever your partial credit for it is. But if as long as you get 80%, then I bump that up to full credit. So that does make a difference. A couple of unit reviews, which apparently I can't spell either, or at least can spell right half of the time. Apparently I was very drunk when I wrote this thing out. Can I still get in there and change it? Nope. Oh yeah, score. There you go. Unit review due on Tuesday, unit review due on Thursday. Your last Physio X is due on Friday the 23rd. Again, I know how much you guys have all been enjoying doing those. And the good news is, well, the bad news is this is the last one. The good news is, though, it has 9 activities, so that should keep you busy for a while. One of the things that in grading your Physio X's from the last part that I want to remind you is first, make sure you do all of the exercises. Make sure you collect all your data. If you do the exercises and you don't collect your data, then it doesn't show up on your lab report. And if it doesn't show up on your lab report, I don't think you've done it. So make sure all your data is there. And make sure also, as I had to remind some of you, that I'm not interested in what information you can cut and paste from Quizlet or some other resource like that. You need to put your answers in your own words. Cutting and pasting answers to reviews from Quizlet is a fast way to get this long activity done, but it doesn't help you learn the material. And it's also plagiarism. So remember that you are responsible for writing your answers in your own words. Please do that. We have one more review. Like the previous reviews, this one is going to be graded for correctness. Like previous reviews, I purposely have not made it available yet. What's going to be slightly different about this review is that there are, as we've talked about up to this period of time, there is a strong relationship between structure and function. We know structure and function go hand in hand. But if you think about it also, in the last section with the muscular system, we also learned the importance of location. Because clearly by drawing the origins and insertions, right, this muscle here in my arm does something different than this muscle here in my leg. Right? One may flex the hip, whereas the other flex the shoulder. Well, it's the same thing with nervous tissue, right? What this part of my brain and this part of my brain do is something different. And while what this part of the brain on one side and this part of the brain on the other side may do the same thing, they can also do different things as well. So one of the things that we are going to be emphasizing a lot in this section is the relationship between location, structure, and function. Now, the good news is if you know one of those 3 things, the structure of our neuron, the function of the neuron, or the location of the neuron, then that should help you to be able to infer what the other 2 is. So that is the nice thing about that. And that's what this review is about really emphasizing that relationship between structure, function, and location. And again, it's due on our second to last lecture day. So we have plenty of time to talk about the anatomy, talk about the structure, talk about the function before we get there. All right, I think you have 2 other assignments that are going to be due on Tuesday, the 27th. One more unit review, you're 14. And then there's also a fun reflex lab that you're going to do as much of as you can at home. But we'll worry about those when we get closer to them. All right, questions on any of that? All right, excellent. So then with that in our pocket, let's move on to our lecture. As I mentioned, we have talked about epithelial tissues, we've talked about connective tissues, we have now talked about muscle tissues. So our last major tissue type we have left to talk about is our nervous tissue. And so that's where we will begin our conversation of our nervous system. As always, let's start with an introduction. What is the function of our nervous system? What is the function of nervous tissue?
Communication.
Yeah, absolutely. As we talked about, it is a system along with the endocrine system that is specialized for communication. And we use that communication, as we've talked about, as that command center for homeostasis to help us to maintain our health and to maintain our balance. And as we also talked about, how does it communicate? What does the nervous system use to communicate?
Action potentials.
Yeah, action potentials. Big, positive electrical signals. Right. We also talked about how the endocrine system also communicates. And remind me again, how does the endocrine system communicate?
Hormones.
Hormones, exactly. And so as we talked about, there are 2 big differences between communication with the nervous system and communication with the endocrine system is the timing. Nervous system is very, very fast, whereas it takes a lot longer for a cell to produce a hormone, release the hormone, diffuse that hormone through our body and have some kind of an effect. But, right, even if I stand the edge of that huge massive pool I have in my backyard and throw a whole big car into it, I may produce a big massive wave, and that big massive wave will spread down the pool, but it also doesn't last very long. So action potentials are a very transient signal, whereas those hormones are much longer lasting, allowing us to coordinate activity over a long period of time. Whereas nervous system allows us to make rapid changes and rapid modifications to respond to our environment and other such. Of course, our nervous, that really only scratches the surface of what our nervous system is able to do for us. Our nervous system is responsible for our perception of our internal and external world, our behaviors, our movements, our memories, our emotions, and so much more that our nervous system does for it. So it's a whole big gaggle of things. And then, of course, the study of the nervous system is the field of neurology or the field of neuroscience. All righty, let's do this way. As we kind of have already hinted at, I think that should be good. When it comes to the functional organization of the nervous system, there are basically 3 main components of it. Anyone have any idea what one of those 3 components might be?
Sensory.
Excellent.
Observing.
Absolutely. So absolutely. So one of those is, of course, our sensory. And what do we mean when we say sensory?
Our eyes and looking around, perceiving things.
Okay, exactly. I like that. So this is where we receive information. That can be both internal and external information. And then once we receive that information, what do we do with that information?
Process it.
True, but we don't necessarily process it at that sensory location. We have a second portion. Again, if you think back to homeostasis, what we referred to as our command center. But here, from a functional standpoint, we can think of it in terms of integration. You've got absolutely the right idea. Integration is where we get the information. We process that information. We compare, and part of that processing is comparing it to our memory. And then once we've done that, we make a decision based on that information. All right. And then once we've made that decision, what do we do next?
Act on it.
Right, exactly. And to act on it, we need some type of motor function. Motor function is where, actually, I guess we got to leave function out of there, is where we effect some change in our body. And of course, to do that requires effectors. And I think we talked about this when we were talking about homeostasis. What are the potential effectors in our body? What are the things that can be effectors?
Muscles.
True. So again, let's be more specific. Skeletal muscle. That's one of the things that can be an effector. But as we learned, there's different flavors of muscle. Smooth muscle, cardiac muscle. And wasn't there something else that could be an effector as well?
The endocrine system.
You got the right idea. Well, remember, endocrine system is one of the integration centers, one of the places where we make a decision and send a signal, part of our command center. But you do have the right idea in that one of the other things that can be effectors are glands. All right, we get up and run around the room 16 times, we start sweating to bring the body back into balance. So we have this functional organization where we have our sensory information, and that sensory information is information that is sent inward to be integrated. And then once we receive that information, we make a decision based on that, and that information comes out in the motor to our effectors to affect some change, right? We've talked about this example a couple times, although I think I'm going to need a little, no, I think we can make that work. And maybe I do need a little more room. Bring this down, bring that down, bring that down. All right, well, that's just stupid. Excellent. All right, we've talked about this example. There you are sitting in your chair, and a bear with an axe walks in the room. You see the bear with an axe. That information travels into your integration center where you process that information. Bear, bad. Axe, bad. Bear with axe, very bad. So you make a decision based on that. You have one of two choices. You either have to fight that bear with an axe, or you have to outrun somebody in the room with you. So you send that signal to your effectors, your skeletal muscles, your glands, your smooth muscle, your cardiac muscle, all of those things, so that you can either run or fight. All right. And those are the functional organizations. Now notice sensory information goes in towards the integration center. And we do have a special name for that direction of information. Information that comes into that integration we call afferent. And then the motor information coming outward, how do you think we refer to that? If afferent is going in, what's coming out?
Efferent.
Yeah, efferent. And yes, I know it's pronounced efferent, but the afferent and efferent, just like AB duct and AD duct, help me remember which is which. There you go. So we have the directionality of that information, we have the type of information, and we have that aspect as well. All right, excellent. However, as has already kind of been hinted at, there are different types of sensory information. Right? Some of the sensory information, as you guys talked about, are things that we can receive from our body, what we call somatic sensory. Somatic sensory is our general sensations. Like, for instance, touch. These are things, external stimuli from outside of our body. Touch, temperature, tickle, itch, pain, right? You can feel, if I take a pin and poke you pretty much anywhere on your body, you are going to feel that. So that is an example of a somatic sensory information. Conversely, as someone mentioned earlier, vision. Can you perceive light from all parts of your body?
No.
No, only from the retinas of your eye. And this is not something you should do. Again, I'm repeating, do not do this. If you do this, I will take away participation points from you in the class. But theoretically, when you wake up in the morning and you're rubbing your eyes, if you were to rub your eyes too vigorously, which I'm telling you right now, do not do, but if you were to push on your eyeball, what would happen? Anyone know? Anyone get a little bit too vigorous with their wiping away those eye boogers in the morning?
It's black.
Yeah, you see when you push on your eye, which again you should not do, you see a light. Not because your hand is suddenly producing light, but your retina is specialized to perceive light. When you push on the eye, what ends up happening is you increase your interorbital pressure, which is bad for your retina, which is why you shouldn't do it, but it pushes on the retina and it stimulates those retinal cells. When they get stimulated, they send an electrical signal to your brain, an action potential to your brain, and your brain thinks it's perceiving light because the retina is stimulated. So not only do we perceive light from just one location, but that one location is specialized to just perceive light. This type of information is what we call special sensory information. This is basically some type of specific input from some type of specialized receptor. Like our ears, like our smell, like our taste, like our vision. Those are our special sensory information. And again, notice this is also external. But what about what's going on the inside of your body? Do you, let me say this, how many people right now know how much calcium is in their blood?
A lot.
Okay, but do you know the precise amount?
No.
Do you know if it's a little high, a little low, just right? I'm about a quart low on calcium right now.
No.
No. However, do you think it is important to know how much calcium? Do you think there are parts of your body that absolutely know how much calcium is in your blood and is doing something about it?
Absolutely.
Or your internal temperature, or the pH of your blood, or how much glucose you have in your blood, and so on and so forth? Absolutely. This is not information we are consciously aware of, but we absolutely positively are monitoring what's going on the inside of our body. And to do that, we have special type of internal receptors that we call enteroreceptors. And these enteroreceptors basically monitor the internal conditions of the body. So these enteroreceptors are monitoring what's going on the inside of our body, sending that information to our command center, to our integration center of our brain, so it can be processed so we can make any changes necessary. So notice we have 3 subdivisions. In the sensory component of our nervous system. And not surprisingly, if we have subdivisions of our sensory, we have subdivisions of our motor. Anyone know what those subdivisions might be? Or how many at least? How many subdivisions?
3.
3? 3 would be a good guess, but if you were a little bit clever, you might have looked at the way that I organized the effector. 2. There you go. Excellent. We have 2 subdivisions to our motor functional organization. And not surprisingly, those 2 subdivisions are determined by the effectors. Effector 1 is skeletal muscle. This is obviously our voluntary control. Of these effectors. And if you remember, we actually talked about this in our muscular system. This is our somatic motor nervous system. So our somatic motor nervous system, I should probably switch these around. Somatic motor nervous system provides that voluntary control of our skeletal muscle. Notice our smooth muscle, our cardiac muscle, and glands are not controlled voluntarily. And what did we call the branch of the nervous system that controlled them?
Autonomic.
There you go. Our autonomic motor, which provides the involuntary control of our smooth muscle, cardiac muscle, and glands. Now, there are actually 2 further subdivisions of our autonomic motor. Any idea what they might be?
Rhythmic.
Say again?
The autorhythmic.
True, but if you think about it, autorhythmic is something that cardiac muscle does for itself. So that is actually not a part of the nervous system. But remember, we do have the ability to control those autonomic, or at least modify those autonomic cells. So again, if I rip the heart out, put it in a petri dish, it'll happily beat away. But I can make the heart beat faster or slower with my autonomic nervous system. And there are 2 branches that have 2 little catchy rhyming mnemonics to help us to remember them. Anyone know? I know, you just may not know you know. What are the
sympathetic and parasympathetic?
There you go. Sympathetic and parasympathetic. And what are the 2 cutesy little mnemonics to help us to remember those? What's the cutesy mnemonic for sympathetic? It's the one usually people know. Fight or
Flight.
There we go. And parasympathetic? Rest and digest. Excellent. All righty. So here we have the functional organization of the nervous system. Questions on that? All right. We need to talk about the structural organization, but before we can talk about the structural organization, it is probably important that we talk about the structures of the nervous system. Vocabulary, after all, is very, very important. So let's talk and identify the structures of the nervous system. What are some of the obvious ones that should jump out right away?
The brain,
There you go. What else?
Nerve.
spinal cord,
Yeah. Excellent. So I think those are the two that I think everyone can come up with right away. That's awesome. Although I love what I also just heard. I think, Elvina, are you the one who said it, or Emily? Someone said it. Nerves? Someone say nerves?
Yeah.
Excellent. Nerves absolutely are a structure of the nervous system. But can someone tell me what a nerve actually is?
sensory cell,
So not exactly. It can be, but here's the trick. So I'm going to erase nerves. We'll come back to it in a second. Let's go back. If you remember correctly, where can I?
like what is it made of?
Yeah, that was kind of what I was looking for. But let's go ahead and do this anyway. I know sometimes the bottom. Do you see that whole circle?
Yeah.
Excellent. Do I have any room to move it down, or is that at the bottom?
You can move it down about half an inch,
There.
a little bit more.
Excellent. Okay, perfect. If you remember, we've seen a neuron. We know that a neuron has a cell body and little dendrites. And then that one big process that comes off, that is the axon with its specializations at the end. So nervous tissue is primarily made up of one of two things. It is either the cell body and dendrites, or it's the axon. So basically it has two components to it. It turns out what a nerve is a bundle of axons. So when we talk about nerves, what we mean when we say nerves is a bundle of axons. And it turns out nerves come in two flavors. Cranial nerves and spinal nerves. And what do you think the difference between a cranial nerve and a spinal nerve is?
Location, location, location.
Exactly. Cranial nerves come off what?
The brain.
Exactly. They come off the brain. And spinal nerves?
Off the spine.
Spinal cord. Excellent. Just out of curiosity, does anybody know how many cranial nerves we have?
12 pairs.
Excellent. 12 pairs of cranial nerves. You will learn everything you ever wanted to know and more about the cranial nerves. And depending on how far behind we get in this section, because we always fall behind in this section, your cranial nerves could be as much as 25% of your lab exam. So we're going to spend a good couple hours talking about your 12 cranial nerves and everything you ever wanted to know and more about them. And it'll definitely be somewhere around 15 to 20%, but depending on how far behind we get on the autonomic and sensory stuff, it could be as much as 25% of your lab exam. So on that day when we're doing it, and as of right now, it looks like that day will probably be Thursday the 22nd. But again, we'll see where we are on things. All right, excellent. Any idea how many spinal nerves we have? Turns out 31 pairs of those. Again, not as fancy, but those are definitely things we need to know. There's one other component we need to really understand what a nerve is. And for that, we have to come back over here to our picture over here. In our picture over here, there's obviously a relationship, as always, as we know, between our functional organization and our structural organization. And there are two branches to our structural organization of our nervous system. Does anybody happen to know what those two branches are? Okay, let me rephrase that. I know you know what the two branches of the structural organization of our nervous system is. Can anybody figure out what they might be? Well, let's jump back for a second. What do we commonly refer to our brain and our spinal cord collectively as? Oops, someone's writing in their chat. There we go, perfect. Central and peripheral nervous system. Absolutely, excellent. Excellent, excellent, excellent. We have our central nervous system. And we have our peripheral nervous system. And of course, is there a relationship between structure and function?
Yes.
Absolutely. Our central nervous system is what is responsible for the processing, for the integration, for the decision making that is going to take place. Whereas our peripheral nervous system is the pathways carrying information into and out of the central nervous system. So these are those afferent and efferent pathways, which again, of course, now we can give a definition to mean that they are going into the central nervous system or coming out of the central nervous system. Now, kind of gave it away already, but what of the structures we've talked about so far comprise the central nervous system?
Right in the spinal cord.
Yeah. I know sometimes this is a little tricky for people because they think, well, doesn't the spinal cord carry information to and from the brain? Yes, that is definitely one of its functions. It does receive information. It can send it to and from the brain, but it also processes information and makes decisions. Now, does your spinal cord necessarily decide what you want to be when you grow up or even what you're going to have for dinner tonight? No. But our spinal cord does make decisions. And what do we call those decisions that are typically made by our spinal cord?
Reflexes.
Exactly, right? When I put my hand on something hot, that information doesn't go to my brain where I go, hey, my hand is on fire. That hurts a lot. Yeah, that really sucks. We should do something about it. Great idea. Let's go ahead and move it. And then we send a signal back and I move my hand as a result of it. No, when I put my hand on something hot, I pull my hand away before I'm even aware of the fact that I should be cursing. Why? Because the decision to move my hand was a very basic but important one. So it was made as a reflex on my spinal cord. It was actually my spinal cord that made the decision that said, hey, Get that out of there. I know this seems like we've tangented a little bit to do this, but the important reason for that is we need to come back to our definition of a nerve. A nerve is a bundle of axons, but now we can add to it in the peripheral nervous system. It turns out there are also bundles of axons in the central nervous system, and of course, because anatomists hate us, and because of the anatomy of it, there is a different name for those, and those are typically called tracts. So tracts is a bundle of axons in the central nervous system. So our nervous tissue, our neurons are made up of cell bodies and dendrites and our axons. And our axons either form nerves in our peripheral nervous system or they form tracts in our central nervous system. That also means that our cell bodies and dendrites I'm just going to put cell bodies, but remember wherever the cell bodies are, the dendrites are as well in our nervous tissue. And guess what? They're going to have different names in our central nervous system versus our peripheral nervous system. In our central nervous system, there are basically two names for the clusters of our cell bodies. The first is because on our nervous tissue, it forms the outer surface. It is known as the cortex. These clusters of gray bodies also may be what you've heard of before as the term gray matter. Whereas our nerves and our tracts are our white matter. So actually, let's cheat. Move this down. This stuff up here is our white matter. Why might the axon, oh, actually, we don't know that yet. We'll get to that. Never mind. Our axons are our white matter. Our clusters of cell bodies are the gray matter. In our central nervous system, it is either called the cortex or bundles outside of the cortex are what are known as nuclei, singular being nucleus. And in our peripheral nervous system, anybody know what we call a cluster of cell bodies in the peripheral nervous system? It's okay if you don't. We're learning all of this vocabulary. Ganglion, which is the singular, although I guess I should, for uniformity's sake, do that. Ganglia is the plural. Oops, not how you spell ganglia. Ganglia is the plural. Singular is ganglion. So, structures in the nervous system, like the brain and spinal cord, like the nerves, like our ganglia, are primarily made up of clusters of cell bodies and clusters of axons. We can also combine these in the peripheral nervous system to form these elaborate networks of nerves. And these elaborate networks of nerves and ganglia, we give a special name to. We call it a plexus. So a plexus is this elaborate combination of networks of nerves and ganglia. This term plexus is something we will identify and talk about more. Again, there are these big elaborate clusters. One of the most elaborate is actually one that is known as the enteric, which is what we commonly and lovingly refer to as our gut brain. As we talked about, and I think we mentioned this briefly earlier in the class, you have that nice big cheeseburger for breakfast, and the cheeseburger goes to your stomach. And then do you have to consciously tell your stomach, hey, stomach, time to get to work churning and mixing up that food?
No.
No. What actually happens is your stomach, when that cheeseburger reaches it, stretches. There are sensory receptors there in your stomach that measure that stretch. And they send that to the enteric plexus, that elaborate networks of nerves of the stomach, where that network, that plexus, actually makes the decision. Hey, the stomach's being stretched out. We have food inside of us. We need to process it. So it perceives that information, makes a decision off that information, and sends that signal to the stomach to start the stomach working. And all of that happened right there in the stomach. The stomach actually made its own decision of how it was going to deal with that food entering it. And so this big elaborate enteric plexus, which we commonly refer to as the gut brain, because it made its own decision for it. And then, of course, we can't forget about our last sensory structure. And those, of course, are our sensory structures. We talked about some of the general sensory structures like the hair root plexes, the lamellated corpuscles, Pacinian corpuscles, Merkel discs, all those kind of things, and then also our retina in our eye and our hair cells in the ears and all of those other things as well. So those are the structures of the nervous system. And as we've kind of already implicated, the brain and the spinal cord are the structures of the structural organization found in the central nervous system. And so that means over here, our peripheral nervous system is, of course, going to be comprised of the nerves, the ganglia, and the plexes, and of course, the sensory structures. So all of those are the components, the structures that make up our peripheral nervous system. All right. Questions on that?
Would the nose or any of that be a part of that sensory, like for smell?
So yes, in our nose, we have special chemoreceptors that are responsible for our perception of smell. So that would be part of those sensory structures that would provide that sensory information. Absolutely. Taste buds on our tongue, right? The photoreceptors of our eyes, the hair cells of our ears. All of those are special sensory structures that allow us to perceive sensory information and then send it in that afferent pathway to the brain. So all of those would be part of the sensory structures. All right. So there you go. Just that easily, we have identified the structural organization, the functional organization, the different components of the functional organization, and the different structures in the structural organization of the nervous system. We've done it here all pretty on the board, but let's go back to your textbook. Again, as we mentioned, from a functional standpoint, we have our sensory function, our integrative function, and our motor function. Sensory receives the information, sends it in. Integrative processes that information. Motor is then what affects the change to bring us typically back into balance. I like the more colorful example of a bear with an axe. Your book uses a more benign example of seeing a glass of water, realizing you're hungry, and then thirsty and taking a sip of it. We also talked about the anatomy. Primarily, nervous tissue is made up of those neurons that we've seen and drawn. that, again, as we talk about, have a cell body and a dendrites, and then have an axon that comes off of it. So the structures are primarily comprised of these two things. These glial cells are basically, for right now, what we're going to refer to as support cells that help to assist the neurons in doing their jobs. But it is the neurons that define the anatomy, like the brain, like the spinal cord, like our 12 pairs of cranial nerves. Again, a nerve is a bundle of axons, and our spinal nerves. Our ganglia, which of course, remember, as we mentioned, is a cluster of cell bodies in the peripheral nervous system, and our sensory receptors. So these are some examples of some of the structures. Not all the structures. Our list had more of them, so make sure you're familiar with all of those structures on the list. But what I really like is while we put it together in words, oh, and then the plexes, can't forget about those, enterics and others. We also have those different functional organizations. Structural organizations, our central nervous system, integration and command center, brain and spinal cord, peripheral nervous system, our lines of communication, carrying information in and out, the nerves, ganglia, sensory receptors. And then, like we talked about, we have subdivisions for our peripheral, our afferent coming in, our efferent going out. 3 subdivisions of our sensory neurons: somatic sensory, autonomic or visceral sensory, and our special sensory. And then for our motor, we had the 2, somatic and autonomic. And then under the autonomic, we had sympathetic and parasympathetic. So again, just like we did it on the board, here we have it in the words. But as I mentioned, what I really like from your textbook is this right here. Notice here they do this excellent job of showing us this organization visually. Here we have our central nervous system, where we're processing the information and making sense of it. And again, it is comprised of the structures that are the brain and the spinal cord. We have our peripheral nervous system, which are the pathways, efferent pathways, sensory coming in, like our somatic sensory from the skin, our visceral with those enteroreceptors carrying the information in. And notice what we're missing here. So let's go ahead and do them the service of giving them something like an eyeball, because after all, there is nothing more important than an eyeball. And that eyeball then can now carry our special sensory information inward. So we can't forget about our special sensory. There we go. Then lastly, notice we have our motor or efferent pathways, including our somatic, where the information goes out to our skeletal muscle, giving us that voluntary control. And our autonomic, where we have that involuntary control of our cardiac muscle, of our smooth muscle. And notice they could have also put a gland in here for us if they had been so inclined. And notice again, we have our sympathetic, our fight and flight, where we're mobilizing resources to deal with stress, or our parasympathetic, our rest and digest, when we're conserving or storing energy, right? Or like what we like to think of as housekeeping type of functions when at rest. All right. So here we see that nice visualization of this relationship between structure and function. And notice we've already started hinting at this by talking about places like the brain or the spinal cord or the eye and the skin. Remember, we also are going to need to add location to our relationship of all of these things as well. But this is our structure and our function. Questions on this?
Nope.
All right, excellent. I will go ahead and leave this picture up. It's a teeny bit early, but this is a good natural stopping point. We've got a fair amount of material to cover today, so let's go ahead and take our first break here. We'll take a 15-minute break. Looks like that means we will restart at 9:06. Oops. And I will start the recording at that point. All right, I will see you guys in 15 minutes. Do you want to stay on this page or do you want to go back to the one that I drew? Which would you guys prefer to look at for 15 minutes?
This one works.
What?
Oh, so this one works.
Okay, perfect, excellent. All right, excellent. See you guys in 15 minutes. All righty. Now that we've talked about the organization of the nervous system, let's get more into the nervous tissue like we said we were going to. As I hinted at earlier, nervous tissue is made up of billions of neurons and trillions of glial cells. And the term glia and neuroglia are terms that are used interchangeably, and I will use them interchangeably as well. They mean the exact same thing. Let's talk about their functions. As we mentioned, the neuron is the functional cell. This is the one that is responsible for the communication. All right, and of course, that communication is the production of action potentials. It is able to do this because it shares some of its characteristics with muscle tissue. Or probably more the other way around. Muscle tissue shares some characteristics of neurons. They are irritable. Remember, this means that they can receive a stimulus and respond to it. Neurons are electrically excitable, which remember means that they respond by producing a big electrical signal. And as we talked about, that big electrical signal is an action potential. Which remember is a big positive signal. And like the neuron, they have conductivity, right? Here, normally I'm sitting in the classroom, and again, I have to work on my analogies, and I pick up my wooden meter stick, which has a nice metal end tip at the end of it. And if I were to take that metal tip and shove it into the light socket, we'd get some real pretty sparks that would occur and some smoke and things like that. And me holding it to the other end of that meter stick, do I have to be concerned about that? Yeah or no? Why not?
A wooden?
Yeah, it's wooden.
It's not conductive.
Exactly. That wood doesn't conduct an electrical signal. Now, if it was a fully metal meter stick, then when I plugged it in there, yeah, I would have that problem. And the same way that we saw the sarcolemma, we had that specialized plasma membrane that was able to produce and propagate, or remember, produce and spread. spread here, but I like the fancy word propagate. That electrical signal, our neuron has conductivity as well, which is why we can take that signal from our small of our back all the way down to our big toe, or vice versa. You step on a tack, that information travels all the way up to the brain to tell you that you've hurt your toe. Excellent. Now, the neurons are the functional cells. They're the ones that are responsible for producing these electrical signals, and they need help. And that help comes in the term of our neuroglial cells. And certainly we will talk about our neuroglial cells. We know our neuroglial cells are responsible for helping the neurons do their job. However, you will notice there is this little asterisk that we have put around or next to the support cell, because in the past few years, there's been some really interesting research that has been done. One of the main drives in this field of research is an individual by the name of Albert Einstein. Anyone here heard of Albert Einstein before?
Yeah.
Okay, excellent. A couple of you have. For the rest, read more. But Albert Einstein, as you may or may not know, was an incredibly brilliant individual who was able to make amazing leaps in cognitive thought that us mere mortals couldn't. And so when he died, he donated his brain to science. Once the scientists finally got a hold of his brain, which is a whole different story that we don't have time to go into here, they were all a slather to be able to cut into his brain and look at what kind of amazing neural connections he must have that are so different from mere mortals like you and I, that allowed him to make these tremendous leaps in thought and understanding. And when they cut up and looked at his brain, what they found out is that he basically has the same number of neurons and the same arrangement of those neurons as you and I, which is good news, because then you can call grandma up on the phone and say, hey, guess what, grandma? I have the exact same neural network as Albert Einstein. And she will be incredibly impressed, and she will send you 20 bucks in the mail. Now, while he had the same number and arrangement of neurons as you and I, his brain wasn't identical to a typical neuron. There were some parts of his brain that had as many as 4 times as many neuroglial cells than a typical brain does. So findings like this and additional studies have started this whole new field of study where we're looking at neuroglial cells in a different way and seeing how they may play an important role in our processing of information. So should you take this class 10 years from now, and again, I don't know why you would want to take this class 10 years from now, but if you would, we may be talking a different story about neuroglial cells by then. But for now, we'll focus on what we know that they definitely do, and that is to provide that support for the neurons. All right. With that, let's talk about the anatomy and function of these cells, starting first with our neuron. And as we've drawn a couple times now, thanks to our discussion of the nervous system, we have here a typical neuron. As we've talked about, this typical neuron has a cell body, or what we refer to as a soma. And one of the interesting things inside of this cell body, when they first were looking at this, there was a particular stain called a Nissl stain, which is a somewhat purplish stain. That when they stained the cell body of our neurons, they were darkly compacted with this massive amount of purple stuff found inside of the neuron. They weren't 100% certain what it was, but it was shown most obviously by this Nissl stain, and so they started calling it the Nissl bodies. However, as our microscopes got better, we started to learn that these Nissl bodies were actually comprised of thin, flattened membranous sacs that are interconnected and had these globular structures lining the outer surface. What does that sound an awful lot like?
Rough endoplasmic reticulum.
Exactly. It turns out that neurons have a massive amount of rough, and I'm going to cheat and abbreviate it here, endoplasmic reticulum. Someone remind me again, what does the rough ER do?
Protein synthesis.
Yeah, it makes proteins. It turns out neurons, remember as we talked about, release neurotransmitters. So those are a ton of proteins that this has to say. And it also has a massive amount of cytoskeletal structures. Or it has an elaborate cytoskeleton, which, as we know, are also made of proteins. So, not surprisingly, neurons need to make a massive amount of proteins, so they have a massive amount of rough endoplasmic reticulum. That makes sense. Notice it has all the other stuff that we would expect a cell to have, stuff like a Golgi apparatus and mitochondria. There's some smooth endoplasmic reticulum, all the kind of things that we expect inside of a typical neuron. But as I also mentioned, it has a very elaborate cytoskeleton. It has all these dendritic trees that need support and stabilize and hold it in place. It has this massively long axon, like we've talked about, that can be over 1 meter long. And it needs to be able to transport those proteins that it makes down to the synaptic end bulb. So it has a very extensive, very elaborate cytoskeleton. And because anatomists love to name everything, the proteins that form the cytoskeleton in a neuron are called neurofilaments. Right. So again, it's in a name, but it's what they are. They are the proteins that make up the cytoskeleton. That is our neurofilament. So it's a very typical cell when we just look at the cell body with more endoplasmic reticulum and more cytoskeleton. However, the biggest difference between a neuron and a typical cell that we've seen are the processes. And as we know, there are two types. We have our dendrites, which, as we see in a typical neuron, are multiple, 2 or more. They are short, elaborately branched, and their job is to receive information. A single neuron in our central nervous system can receive information from as many as 10,000 other neurons. So a single neuron could have 10,000 synapses on it, where it is receiving that information, and that is what those dendrites are. Remember, we have a second type of process. That second type of process is an axon. Neurons have one and only one. It is long, it is unbranched. It carries information away from the cell body. And there's one more very important piece of information we need to talk about for the neuron. For the axon. The plasma membrane of the axon is specialized to produce and spread an action potential. Think of the implications there for a minute. What that means is that the dendrites and cell body cannot produce an action potential. Only the axon is capable of producing and spreading an action potential because it has a special plasma membrane. And because it has a special plasma membrane, do you think that special plasma membrane gets a special name?
Certainly.
Absolutely. And the special name for that is axolemma, right? Sarcolemma in our muscle cell, axolemma, because it is that specialized plasma membrane of our axon. All right, with me so far? All right, so let's think about the implications of this. Our plasma membrane of our axon is specialized to produce an action potential. Our plasma membrane of the cell body and dendrites isn't specialized, not capable of producing an action potential. So there has to be some transition region. Some region where the plasma membrane changes from specialized to not specialized. Or really, in this case, because if we think of a directionality, from being non-special to being special. This transition region is what is known as the Axon Hillock. So here in the Axon Hillock, we have this transition region where it's slowly going from blue to red. So we've got this purple region in between. All right, we'll talk about what the specialization is in just a minute, but let's further take this one step further in terms of implications. Right here at the very beginning of the Axon Hillock, do you think our plasma membrane is specialized enough to be able to produce an action potential?
No.
No. And what about right here at the end of the Axon Hillock? Do you think
No.
there it's specialized enough?
No.
It turns out it is. So here, no. Here, yes. Right next to the axon, it is specialized enough, right? 99%, yeah, it's going to be specialized enough. 0%, 1%, not enough. 99%, yes, enough. So that means somewhere in the middle here, somewhere here in the middle, oh, no, I didn't want that to be purple. Somewhere here in the middle of our axon hillock is an area where our plasma membrane has become changed enough where it can now produce an axon, an action potential. And this area inside the axon hillock, where the plasma membrane is transformed enough to produce an action potential, gets a special name. Of course it does. And that special name is the trigger zone. Now, looking at it with the naked eye or even with a light microscope, can we see where the trigger zone is?
No.
No, of course not. But we know that somewhere inside of this axon hillock is the trigger zone. And that trigger zone is going to be where we're able to produce our first action potential. All right. Questions on that?
Negative.
Excellent. So, as we mentioned, we have that axon hillock with the trigger zone inside of it. And those are the specializations at the proximal end. The good news is we've already learned the specializations at the distal end. Again, there is much terminology associated with this. I will use the terms that I have been using for consistency's sake, but as always, you may use any appropriate terminology. When that one single axon starts to branch, we call these branches axon terminals. Oops. Notice your book here uses the term telodendria. And at the end of our axon terminals, we have what we called our synaptic end bulbs. Your book here uses the term synaptic terminals. These are also referred as the synaptic buttons, terminal boutons. There's also, like I said, there's probably easily a dozen terms that have been used for these synaptic end bulbs. We'll use synaptic end bulbs because it's consistent. And as we've also learned, there are specializations to the synaptic end bulb. What are the specializations we've learned about for the synaptic end bulb? Come on, I know some of you had this as an essay question on the last exam.
Exocytosis of acetylcholine.
Okay, so you're right. We don't have to worry about from the function, but you have that right idea. Absolutely, we have vesicles containing, and again, in this case, because we could be talking about any old typical neuron, we'll use the generic term, a neurotransmitter. Absolutely. And you're right, our goal is to exocytose them. So what did we use for that process of exocytosis? What triggers exocytosis?
Calcium.
Proteins.
Yeah, sure, absolutely. We also have lots of proteins that we know are going to move the vesicle. But then again, what triggers that is our calcium. So we're going to have voltage gated calcium channels to let that calcium in. And remember, this is an active process, so we also have lots of mitochondria. Right. No new information there. That's stuff that we already knew about how a typical neuron communicates with a typical cell. All right. Questions on that? Excellent. All right, but notice we have to come back to this pesky word typical. What does that imply?
It's a non-typical neuron.
Exactly. You are absolutely correct. Exactly. What that means is that there are different types. And actually, let's do it this way. And not surprisingly, we can classify neurons one of two ways. What are the two ways we can classify neurons?
Bipolar.
Well, okay, so structural classifications.
And functional.
There you go. Excellent. So, as usual, let's draw a line down the middle. And once we've drawn that line down the middle, we can talk about our structural, and I think I can do that. Let's put it here for now, and then we can move it as needed. Let's talk about the structural classifications first. And it turns out there are 3 structural classifications. And with our structural classifications, the neuron is defined by the number of processes coming off the body. Now, someone just mentioned bipolar. With a name like bipolar, guess how many processes it has coming off of the cell body? How many tires does your bicycle have? Oh, there we go. Samuel's got it. 2. Excellent. So let's draw this. This, of course, starts with a centrally located cell body. And off of that centrally located cell body, we have one set of dendrites. Now, as we've seen, just like axon, these dendrites can branch. And we have one axon, which, as we know, can branch to form axon terminals. So notice for this, the direction of information is down the screen. All right, so our info is traveling down the screen. And we have this bipolar neuron that has 2 processes coming off of it. So one dendrite, one axon, and then a centrally located cell body. All right, we comfortable with that?
Yep.
That is what I like to hear. Excellent. So let me draw a different one now. This one here has a cell body, like all of them have a cell body. But this cell body is off to the side. It is a peripheral cell body. Cell body that is off to the side. And coming off of that is a single axon. Now, as we know, axons can branch at the end, give us our axon terminals. We still need to be able to receive information. We still need dendrites, but in this case, the dendrites actually connect directly to the axon. So notice our information is still going down the screen here. But in this case, the only thing coming off of our cell is a single neuron. Based on this arrangement, guess what we call this type? Oops, hold on. Black.
Unipolar.
Exactly.
That'd be like the ones that are like involved in like reflexes and things, because it looks like it doesn't have to go.
Well, so is there going to be, do you think, a relationship between the structural classifications and the functional classifications?
Probably, yeah.
Yeah, absolutely. So we will do that, but let's talk about them first. So with unipolar, only one process coming off of that And so we have that there. Excellent. I do. Perfect. Notice, however, neither of these look like what we called a typical neuron. A typical neuron, remember as we saw, had a centrally located cell body. Wow, that's huge. It had 2 or more dendrites coming off of it. And then, of course, our 1 axon. Again, notice despite the difference in anatomy, all of them carry the info in the same direction. Oops, this should say 1 axon. Excellent. So not surprisingly, this is our most common type. And since this one has 3 or more processes coming off of it, guess how we identify it?
Multipolar.
Multipolar, exactly. There you go. These are our 3 structural classifications. Bipolar, that has 2 processes, unipolar, that has 1 process, and multipolar, that has 3 or more processes. Now, because your book uses this term, I will mention it briefly here, but feel free to ignore it afterwards. If we take a close-up view at this unipolar neuron, from a developmental standpoint, what actually happens for some of these is we have 1 bit of axon that grows out that way and 1 bit of axon that grows out that way, that as this cell matures, they kind of fuse together into, nope, didn't want that. They kind of fuse together into 1 structure. Because of that, sometimes they will refer to these unipolar by the term pseudo unipolar. Like I said, it's not an overly important term. We will use the terms interchangeably because from a functional and structural standpoint, they work exactly the same way. But your book does use the term, so I wanted to at least explain why that occurs. It's strictly from a developmental standpoint. You actually have 2 branches that go off and grow in the opposite direction, and then they basically fuse together. So technically, it starts as 2 processes and not 1 process, but it fuses together in 2. And because our anatomists hate us, they decided to sometimes refer to these as pseudo unipolar. But again, you're not going to be required to use that term. You may simply use the term unipolar, although pseudo unipolar is fun to say, fun to spell. So if you want to do that, you're welcome to do that. All right. Questions on that? All right, excellent. We have now done our structural classifications. Let's switch and do our functional classifications. Our functional classifications, and it turns out there are also 3 of those, are basically determined by the direction they carry information. So if you think about that, one way that they could carry information is into the central nervous system. We would refer to this as afferent information. And what kind of information do we carry afferently?
Visual.
True, that's an example, but what would the generic term be?
Sensory.
Sensory. So one functional classification is sensory, or again, what we refer to as afferent, from its direction, carrying information into the central nervous system. So if sensory, what is one of the other functional classifications? Well, think back to our functional divisions of the central nervous system. If we have afferent coming in, what else do we have?
Efferent.
Efferent going out. And what kind of information is efferent going out? Motor information, remember, is what goes out. So this carries information out of the central nervous system. I don't know why I have central twice there. That bothers me. Can't fix that, so I'll cheat, do that. Excellent. So our third functional classification is basically, its job is to carry information between our sensory and motor. Or we could also think of this as to carry information within the central nervous system. And the types of neurons that carry information between the sensory and the motor, the ones that carry the information within the sensory nervous system, are what we call the interneurons. So we have sensory neurons, motor neurons, and interneurons. Of these 3 types, which do you think are the most common functional classification? Where do you think we have more neurons? More sensory, more motor, or do we have more neurons in our brain and spinal cord?
Brain and spinal cord. Interneuron.
Yeah. Absolutely. The vast, vast majority of our neurons are found in the brain and spinal cord. So our interneurons are the most common structural type. Now, as we know, there must be a relationship between the structure and the functional classification. If neurons are the most common structural classification of neurons, what do you think, of functional neurons, what do you think the structural classification of our interneurons are?
Multipolar.
Yeah. Excellent. Unipolar are most of the sensory neurons. Now there's that pesky word again, most. Most, of course, means not all, because there are some neurons that are associated with our special sensory structures, like our retina for our eye, like our inner ear, which gives us our hearing and balance, like our sense of smell. It turns out that only our special sensory structures have bipolar neurons. So just a few special sensory structures have bipolars. Most of the sensory are unipolar. And that leaves us with motor. Well, it turns out that all motor are also multipolar neurons, because after all, multipolar neurons are our most common structural classification. So those are our three structural classifications, those are our three functional classifications, and this is how they relate to each other. Right? Our sensory, most of them are unipolar, and special sensory contain bipolar. All motor neurons are multipolar, and all interneurons are multipolar. So there you go. Our relationship between our structural classifications and our functional classifications. And of course, I've done an amazing job drawing this, but we do have some pretty pictures from your textbook. Notice here, we see an example of a very nice bipolar neuron, single cell body, one dendrite coming off of it, one axon coming off of it, two processes. And again, these are associated with our special senses, like our retina, like our inner ear, like our olfactory region, our sense of smell. Unipolar, notice its cell body is off to the side. A single neuron carrying the information down where it's connected to the side. Dendrites connect directly to the axon. And these form the majority of our sensory neurons. And there's our, what we refer to as typical, and by far the most common of our structural classifications. And this looks like we looked at before. Multiple dendrites off a centrally located cell body, one axon, ending with synaptic end bulbs. And again, these form all of our motor and all of our interneurons. Functionally, the direction they carry the information, motor or efferent, which are multipolar, sensory, afferent, mostly unipolar, interneurons, making up over 90% of our neurons, are multipolar. So, three structural classifications, three functional classifications, and that's it, right? Well, I'm asking the question, so what's the obvious answer? No, of course not. No. Is there only one type of interneuron or only one type of multipolar neuron or something along those lines? No, there are dozens of different types. Let's look at two examples. One of the ways, again, remember, one way we can distinguish neurons is by their location. So even though this is something we haven't talked about at all, if I tell you that one type of multipolar neuron is an anterior gray horn cell, Guess where you would find the anterior gray horn cell?
The anterior of the brain.
Well, in the anterior gray horn, which happens to be in the spinal cord, actually, but you got the right idea. Some are named by their location. Some are named by their shape, like here, our pyramidal cell. This pyramidal cell is the one that's actually found in your cerebral cortex of your brain, your gray matter. And it's given its name because it has this pyramid-like shape and these very long extending dendrites to receive lots of information out of it. But this nice pyramid shape, triangular shaped cell body is where it gets its name. And then, of course, the third way these get their name is like the Purkinje cell. The Purkinje cell is actually an incredibly beautiful cell. Notice as you look at this, this illustration looks very two-dimensional, like we didn't have a very good artist. It kind of looks like this elaborate tree, but instead of having all the branches going in all sorts of directions, it almost looks like they have the branches going out in this big fan-like structure. Right? Isn't that kind of what it looks like? Well, it turns out that's because that's what this cell looks like. This Purkinje cell has an elaborate dendritic tree that basically just fans in 2 dimensions. So if you took this cell and rotated it to the side, all of the dendrites would all line up with each other. Now, as we mentioned, structure, function, and location happen to go together. These cells happen to be found in a region of the brain called the cerebellum. Histologically, these will be the first cells that we look at under the microscope, these Purkinje cells. And where did Purkinje cells get their name?
Bob Purkinje.
Good old Bob Purkinje. Purkinje is what we lovingly refer to as an anatomy whore. Purkinje was an amazing neuroanatomist who loved the microscope and was an amazing artist. He would look into the microscope, see these beautiful images under the microscope, and he would recreate them in these amazing drawings that he would do and then publish. Again, some of his drawings of these neurons and other parts of the tissues of the body have sold for hundreds of thousands of dollars. They're amazing works of art that he would do. And of course, when he would draw these cells that he saw in the body, he would plant his flag in it. So here in the cerebellum, we're going to have Purkinje cells. When you get to the heart, there are Purkinje fibers that we're going to see there. Again, he's planted his flag all over the body. Again, named all these things after himself. But again, his skills with a microscope and with a paintbrush were truly, truly amazing. So not surprisingly, there are different types of unipolar, different types of multipolar, different types of bipolar, and typically given names by their locations, by their shape, or by the Bob that found them. All right. Questions on that? All right, that is it for the anatomy of our neurons, or the classification of our neurons. And your book's got some great tables that do a good job of showing us this. But what we need to do next is talk about those support cells, those neuroglial cells. Again, we got a fair amount of information to cover, and this is, even though it's a teeny bit early, this is a good natural stopping point because we're covering a lot of material today. So let's go ahead and take our second break here. Looks like that's going to mean coming back at 10:09. And we will restart at that point. And I will remember to start the recording like I forgot to do for the review. So again, hopefully this will help me to remember. All right, excellent. So I will see you guys in 15 minutes. Let's pick up where we left off. We had just finished talking about the anatomy of a neuron and its classifications. So we are now going to talk about the neuroglial cells. there have been over 100 different types of neuroglials that have been identified in the body. Obviously, we're not going to cover anywhere near that many. We are going to focus on the most common types and their function. And another important thing to remember is that there are different glial cells in the central nervous system and the peripheral nervous system. While obviously they may be somewhat similar in their function, the way we're going to distinguish our glial cells are by the ones that serve function and are found in the central nervous system and those in the peripheral nervous system. So if we switch pages and look at this pretty picture here, we can see some of the most common types, and as you'll notice, there are going to be 4 that we will identify in the central nervous system, and then, central nervous system, and there are going to be 2 in the peripheral nervous system. So that is what we're going to look at, just these 6 types. And like I said, of the over hundreds that have been found, these will be our 4 most common and the ones we will talk about. Let's start here with the astrocytes. Astrocytes are the most common type of glial cell found in the central nervous system, which is again where most of the neurons are located. So astrocytes are the most common type of glial cell, period. And if we look at the anatomy of it, we can see somewhat where it gets its name. It has a large centrally located centrally located cell body, and from that it has many processes that come off of it, giving it a star-like appearance. Where it gets its name, it is an astrocyte. Not surprisingly, this helps to form the structure of the nervous tissue. Again, if we were in the classroom, one of the things we'd be doing sometime in the next week or two we would be doing a brain dissection. And when we get that sheep's brain and we do the brain dissection, when you get to feel it, it kind of has the consistency of kind of like a rubber ball to it. The reason it is so firm is because we have put a fixative in it that locks the proteins together, giving them more shape and structure. So it makes it easier to make the incisions and to see the anatomy of it. The actual nervous tissue inside your brain, it has more of the consistency of like Jell-O. Or like a Jell-O jiggle or something like that. So it has a little bit of structure and form to it, but it's very soft and somewhat pliable. And it is these astrocytes that give that structure, that scaffolding for the tissue. Certainly a very important function that it provides. Remember, one of the other things we talked about is that at a synapse, there are enzymes that can break down the neurotransmitter. Some neurotransmitter can be endocytosed back into the synaptic embulb, and some of it diffuses out, which is awesome because it gets it away from the motor end plate, but we don't necessarily want a whole ton of neurotransmitter diffusing through all of the interstitial fluid of the tissue because it might diffuse its way over to another synapse. So these astrocytes play an important role in metabolizing neurotransmitters. So if neurotransmitters are diffusing through the interstitial fluid, they can gobble them up and stop them from affecting any other cells. Super vital important. We hinted at with muscle fatigue that ionic imbalance can have an effect on the function of cells. And as we know, there should be a lot of potassium inside of cells and very little potassium outside of the cells. So astrocytes play a very important role in the reabsorption of potassium, keeping it out of the interstitial fluid, keeping it low in the interstitial fluid so cells can continue to do their function properly. All of these things are vitally important functions that our astrocytes do, but it's not the most important, right? If you look closely as we come back here to our illustration, you'll notice that yes, it's helping to hold and stabilize cells in place like the neuron and its axon. But you'll notice that many of the processes of this wrap around these red things. Any idea what these red things might be that it's wrapping around?
Capillaries.
Yeah, there you go. Exactly. They're on the capillaries. These astrocytes help to form what we call the blood-brain barrier. As we know, blood is vital for carrying oxygen and nutrients and materials like that throughout our body. But it also carries wastes and other materials as well. And do we necessarily want those wastes or other types of toxins getting out of the capillary into our nervous tissue?
No.
Right, because wastes and toxins could damage the cells. But that's okay, we can make more, right?
Negative.
Right, remember, as we talked about, nervous tissue like cardiac muscle doesn't regenerate. So we want to limit what is able to get out of the capillaries into the nervous tissue. And that is a hugely important function. So vitally important function of our astrocytes is to form this blood-brain barrier. However, as we've also learned, as we start to understand how the body works, we can use that to our advantage. Now, this blood-brain barrier is good, but it's not perfect. After all, if you think about it, it is formed by wrapping plasma membrane around the capillaries. And what is plasma membrane made up of again? Primarily?
Phospholipids.
Phospholipids. So something that is lipid soluble, like, oh, I don't know, alcohol. Is alcohol able to sneak its way out of the capillary and because it's lipid soluble, get through that wrapping of plasma membrane and have an impact on our neural cells?
Absolutely.
Yeah, absolutely it does. Absolutely.
Why we have a drink for breakfast.
Exactly. Yep. Exactly. With that cheeseburger, having a shot of Jägermeister is probably not the best thing for comprehension in this class. Absolutely. Maybe required, but it's not necessarily the best thing for comprehension. But remember, as I also talked about, as we learn how the body works, we want to be able to manipulate that to our advantage. For example, how many here have heard of a condition known as Parkinson's disease?
Yep.
Excellent, a handful of you. Parkinson's disease is a neurological disorder. Symptoms are typically associated with a tremor at rest. So when the person's hand is resting, they can have a tremor to it. They also have difficulty with they may try to do an action and may start and stop that action as they're doing this. As I mentioned, this is a neurological disorder that is associated with a decrease in a very important neurotransmitter called dopamine. People with Parkinson's disease lack dopamine, which has a lot of implications on the body, and one of them is many of the symptoms. And what they have found is that if they can increase dopamine in the brains of these Parkinson's patients, then these symptoms go away, which is awesome. But here's the problem. Dopamine doesn't cross the blood-brain barrier. So if you're trying to get dopamine into the brain of a Parkinson's patient, can you give them a pill or a shot in the arm and get that dopamine into their brain?
No.
So how do they do it?
Attach it with a lipid base.
True, that might be one way that they could, because we know lipids can get in, but it turns out it's not that easy to do. So what they do instead is they drill a hole in your head and inject the dopamine directly into the brain. Now, surprisingly, even people with very extreme Parkinson's symptoms are somewhat hesitant about having holes drilled in their head. So is this necessarily a very viable way of getting that dopamine into their brain? No, of course not. However, many pharmaceutical companies worked at this, trying things like Brian suggested of coating them in lipids or finding some other way to sneak them in. But like most chemicals that are present in our brain, do they just spontaneously appear as just a fully complete molecule? No, there are typically steps involved in the process of making them. These components along the way are what we call precursors. And it turns out an important precursor in the making of dopamine is a molecule by the name of L-DOPA. L-DOPA is a precursor that if we introduce it into the brain of a Parkinson's patient, the Parkinson's patient brain will convert it into dopamine. We'll convert it into dopamine eventually. And it turns out L-DOPA will cross the blood-brain barrier. So basically what this means is they can take it as an injection or they can take it as a pill. It will cross the blood-brain barrier, get into their brain, produce dopamine, lessen their symptoms, and no holes are drilled into the skull. Patients are happy, stockholders of the pharmaceutical company are happy, everybody is happy and lives happily ever after. All right, so again, as we've talked about, once we understand how these processes work, we can manipulate them to our advantage. And this is a great example of that. All right, questions on this? So again, like I said, astrocytes, vitally important because they form that blood-brain barrier, but they also provide the scaffolding for the brain. They break down neurotransmitters. They help to regulate potassium balance. And they are by far the most common neuroglial cell found in the central nervous system. Great question. No, they typically don't have anything to do, at least as far as we're aware, with epilepsy. Typically what happens with epilepsy is a problem with the neurons. What happens is neurons in some regions produce inappropriate neural activity. And that inappropriate neural activity can spread like a storm over the brain, causing severe epileptic seizures. Why that occurs is not fully understood. Interestingly, though, it might be because, again, as we talked about, these play an important role in metabolizing neurotransmitters and potassium. So ionic imbalances in neurotransmitters could be something that leads to that. But my guess, again, it's not my area of expertise, but my guess is that those are such obvious examples. And since we don't know what causes epilepsy, my guess is that it's probably not associated with these, at least not from that aspect. There could be something else that they're doing possibly, but as far as I know, there's not a link between astrocytes and epilepsy. Although it's a great place to look. And like I said, I'm sure there are neuroanatomists or neurophysiologists who are doing that because they're such a common glial cell. But as far as I'm aware, there isn't a link. Great question. Awesome. Excellent. Any others on these? All right, let's talk about our next glial cell. These are ependymal cells. Ependymal cells are a neural form of an epithelial cell. Of course, as we know, epithelial cells line surfaces. And there are indeed surfaces associated with the brain. In fact, our brain is both surrounded by fluid and also has spaces inside the brain that contain fluid. And anybody know this fluid that is found both on the outside and actually in spaces on the inside of the brain?
Cerebrospinal.
There you go. Cerebrospinal, all one word. Fluid, which again, as you've heard, I'm sure on any medical show, or if you're abbreviating it for an exam, CSF, right? People talk about CSF. That is cerebrospinal fluid. Absolutely. This cerebrospinal fluid bathes the surfaces. It provides cushion for protection. But it also contains chemical signals, nutrients, and can remove waste from the nervous tissue. So it is a very, very important substance that is produced in and around the brain. And it is these ependymal cells lining the surfaces that help in the production and regulation of that cerebrospinal fluid. So these basically help to make the cerebrospinal fluid and then regulate the composition, helping to remove waste or maintain the ionic imbalance and things along those lines as well. All right. Questions on that? All righty, excellent. Let's talk about our next one. And these are our oligodendrocytes. If we've learned anything in this class, in every section, there's going to be some alphabet soup term that we're going to need to be able to spell. And this is that for our glial cells. Here, we see a large centrally located cell with many processes coming off of it. One oligodendrocyte can have as many as a dozen processes that come off of it. Not quite as many as an astrocyte, but still a lot of processes. And notice what these processes do. They wrap themselves hundreds of times. In this case, notice not around the capillaries, but around the axons of the neurons. When they do this, they form a substance that we call a myelin sheath. So here we see a myelin sheath wrapped around an axon. And notice, because oligodendrocytes have many processes, they can form many pieces of myelin, being able to wrap around as many as a dozen different pieces from a dozen different axons. Here, notice this one, we see forming 3 pieces of myelin sheath around 3 different axons. The question then becomes, why? Why wrap the plasma membrane hundreds of times around these neurons? Well, remember, axons are kind of like the wires in your wall. Their job is to carry an electrical signal from one place to another. So if you ripped open your wall and looked inside, you'd see a bunch of wires. And would those wires be completely bare, where you just see bundles of copper that are all wrapped and bundled together?
No.
Why not? Why?
Well, the reason the reason those are so they don't arc.
Yeah, exactly. When you produce an electrical signal, like for instance, flipping the switch to turn your light on, do you want it to open your garage door and turn on your garbage disposal and do all these other things at the same time? No, we want to isolate and insulate the signal so that when we produce that electrical signal, it just goes where we want it to go. That is important and that is vital. But it turns out it does something even more important and more vital than that. It turns out our myelin sheath increases the speed of our action potentials as much as 6 times faster, meaning that a signal getting from point A to point P can get there much, much faster if that axon is myelinated. And most axons are myelinated. Of course, what's the key word in that sentence?
Most.
Most means not all. There are some unmyelinated axons, but most of the axons we are going to run across are going to be myelinated. Which brings us back to one more thing. Remember, as we talked about, axons form our nerves, and they also form our tracts. Remember, they're nerves in the peripheral nervous system. Oops. And they form what we call tracts in our central nervous system, which happens to be where we are now. And if you remember, our tracts we referred to as white matter. When we look at the brain and the spinal cord, we talked about how the cell bodies and the dendrites form the gray matter, the cortex and the nuclei, and the axons form the white matter, the tracts. And now you can see why. Because most axons are myelinated. And what is myelin made up of again?
Oligodendrocytes.
Yeah, and more importantly, the plasma membrane of those oligodendrocytes wrapped around them hundreds of times. Right, so the plasma membrane is wrapped hundreds of times around the axon. And of course, what is the plasma membrane primarily made up of?
Phospholipids.
Phospholipids, fats. So when you take a fat and wrap it 100 times around something, when you get that nice marbled steak, what color is the fat on that steak?
White.
White. And that's why we get the white matter here. Those phospholipids, those fats being wrapped hundreds of times around the axons, makes the tracts of our axons myelinated, makes them whitish in color. And so that's why it is called our white matter. All right. Questions on that? Excellent. When we talk about our action potential and its propagation, we will talk more about how exactly it is that that myelin sheath increases the speed of the action potential. But at least now we know the anatomy of what makes it. All right, one more class of cells that we need to know in our central nervous system, the microglial cells. Microglial cells are actually essentially similar to white blood cells. They're there for defense. And they're phagolytic cells. What does that mean?
Seek and destroy.
True, absolutely. But in this case, specifically, the way they destroy is by eating. They eat things. When there's a site of an injury, right, you get a big sword cut to the head. They're going to go there to look for any harmful pathogens that may get in, but also any cells of yours that are dead or damaged, they will remove those as well. So what happens is they cluster in an area. When they cluster in that area of the injury, they take care of any pathogens. And as I mentioned, they are going to destroy and eat any dead or damaged cells in that area. Really good, really important. But here's the problem. Can we replace those dead and damaged cells?
No.
We cannot replace those dead and damaged cells. So instead, these microglial cells basically form scar tissue at the site of the injury. And as we know, does scar tissue function the same as the tissue that was there before?
No.
And this is basically what we call traumatic brain injury. There is an A in there. There you go. That looks right. Is that right? Doesn't look right.
Yeah.
We'll pretend it's right. All right, excellent. Causes that traumatic brain injury, that decrease in function. So it's a two-edged sword. Yes, it is definitely helpful, but it also has problems as well. So as we mentioned, one of the things that we're interested about as we learn how the body functions is ways we can manipulate it. All right, I'm going to tell you a story, but I want to emphasize three things about this story. One, I was not involved. Two, it happened many, many, many years ago. And three, I promise you, everybody involved died horrible, slow, painful deaths. Okay? So everybody got what was coming to them. All right, excellent. So at UC Davis, which is one of the major sites involved in the study of traumatic brain injury, there were some researchers there, again, many years ago, all died horrible, horrible, painful deaths that studied rats. And one of the things that rats can do is swim. They're not particularly good at swimming, but they can swim. So what they would do is they would put them in a pool of water to swim. And what they would actually do, submerged under the water, would be an acrylic base. Being acrylic, it was clear and couldn't easily be seen. But what would happen as that mouse, so let's go ahead and draw our mouse. Oops, no, can't have a blue mouse. That's silly. There's our mouse. There's its head. There's its tails. There's its legs. Perfect. And what would happen is it would swim around randomly inside of the pool, and by accident, it would find that acrylic base. And when it found the acrylic brace, it could stand on the brace. It wouldn't have to swim anymore, and he was happy because he didn't have to exert all that energy swimming anymore. So they would do that, and the first time he would randomly bump into it. The second day, when they put him in the pool, guess what happens?
You go to where the base was.
Yeah, he pretty much remembers where the base is, and he's able to swim to it pretty rapidly. And so his time, and then eventually gets to a point, he knows exactly where it is. They put it in, they measure the time it takes for him to get there, and he pretty much is able to swim both straight there, and they measure the time, and he does great. Excellent. Then what they would do is they would take the rat with its head, and they would connect a hollow tube to his head, a standard length hollow tube, where they would then release a standard weight from a standard height that would basically fall down the tube and whack the rat on the head, producing a standardized traumatic brain injury. Again, I was not involved. All these people have died horrible deaths as a result, okay? But they would induce a traumatic brain injury into these rats, a standardized one as best as they could. And when they then put the rat into the pool, guess what happened?
Swim around aimlessly again.
Not quite aimlessly, but they definitely weren't as good as they were before. So with that traumatic brain injury, they actually got reduced function, right? Because they had a traumatic brain injury. Then they did the exact same thing with a second batch of rats. But this time, instantly after inducing the brain injury, they would inject them with a chemical, and that chemical basically reduced or inhibited the function of the microglial cells. So now the microglial cells weren't able to go to the site of the injury to take care of any pathogens, to take care of any dead or damaged cells. And what would happen when they then put those rats into the pool, what they found is that they had significant improvement. There was still a deficient, but these did much better than those without the medication. And what they found out because of this is that while these microglial cells are important immune cells, they're also very aggressive. As I mentioned, they go to an area and destroy dead and damaged neurons. Well, it turns out some of those damaged neurons were damaged enough where they would die, but some of those damaged neurons were actually, if left alone, able to recover. Now, did the microglial cells take the time to decide whether a neuron could recover or not? No, kill them all, let God sort them out, right? So it was pretty indiscriminate. So by suppressing the microglial cells, they actually found improved function. More of the nervous tissue was able to actually regenerate, and well, not regenerate, but recover from the injury. And they found other things as well. Not only is this chemical something that can do it, but temperature has a big effect. Decreasing body temperature dramatically decreases the function of these microglial cells and some of the other key factors that affect the regeneration or the inflammation of neural tissue. And so again, we tell this story, it sounds gruesome, it sounds horrible that we're doing this, but we have gained a tremendous amount of incredibly viable and important information to this that we've been able to take to humans. And one of the places where you see this tremendously is in sports, even as recently as 10 years ago, right? If someone had coming down on a kickoff in a football game, the tackler puts his head down, he gets into that injury to his spinal cord, and basically he's not walking for the rest of his life. Whereas, gosh, 3 years ago, first game of the season for Buffalo Bills, there was a player who was on the kickoff team. Very first play of the game, very first kickoff of the game, came down, got a traumatic injury to his spinal cord, and he was actually playing by the end of the season. Our understanding of how to treat neural injuries has changed dramatically, unfortunately, because of animal research. Right? Again, what they did is not necessarily nice, but we've received and learned a tremendous amount about how the brain works and how we can use that to improve individuals. So, gosh, 25 years ago, let's say you starred in a couple movies as Superman on top of the world. You're riding your horse one day, get bucked off the back, and damage your spinal cord. And you spend the rest of your life in a wheelchair, right? Unable to walk. That's what happened to Christopher Reeves. But in that time, our ability to be able to understand how these traumas affect our nervous tissue and what we can do to improve conditions to limit the overall damage have changed dramatically. All right, so I'll get off my soapbox. Questions on this? Let's go ahead and erase that. All right, so that gives you an idea of just some. Like I said, over 100 different glial cells have been found. Those are the 4 most common. Those are the ones you're responsible for in the central nervous system. You are also responsible for 2 in the peripheral nervous system. And not surprisingly, they're going to be somewhat similar in their functions. What is the structural classification of this neuron that you see right here?
Unipolar.
Unipolar. So what is its function?
Sensory.
Sensory, excellent. We know those 2 things go hand in hand. It turns out, since we're talking about the peripheral nervous system, I will tell you right now that these cell bodies cluster together in a structure in the central nervous system. In the peripheral nervous system. And what do you think we call that structure where these unipolar neurons, cell bodies, might be found clustered together in the peripheral nervous system? What did we call a cluster of cell bodies in the peripheral nervous system?
Nerves.
Cell bodies, nerves or bundles of axons. What did we call a cluster of cell bodies in the peripheral nervous system? Yeah, ganglia. Singular ganglion. Excellent. So when we look at ganglion, and I will tell you in particular, this one, it happens to be found in what is known as the dorsal root ganglion. Guess where the dorsal root ganglion is located? I know you haven't talked about the anatomy yet, but you might be able to figure it out.
Dorsal's the back.
Yeah, so it's on a dorsal root. I know the term dorsal root doesn't mean anything to you yet, but the dorsal root ganglion happens to be on the dorsal root. So we have a location, cells in the dorsal root ganglion. We have a structural classification, unipolar. We have a functional classification, sensory. So if we see the dorsal root ganglion, even if we don't know anything else, we know its neurons are going to be unipolar and they're going to be sensory in function. If I see this unipolar neuron, I know it's functional and I know it's going to be located in the dorsal root ganglion. If I tell you a neuron is sensory, you know structurally it must be unipolar, and that means it's most likely going to be found in the dorsal root ganglion. Structure, function, location, all go hand in hand in hand. So in that dorsal root ganglion, we will see all these unipolar cell bodies. And what you'll notice is that there are these cells that wrap around them on the outside, circling the unipolar cell body, kind of like satellites, which is why it gets the name satellite cells. These satellite cells form these satellites around these unipolar neurons. And they're basically similar in function to the astrocytes. They don't form the scaffolding the same way. They're not helping to hold the tissue together, but they're going to metabolize our neurotransmitters. They are going to maintain ionic balances for the cells. So in this fashion, they're going to provide that similar function to the astrocytes. Help to metabolize any neurotransmitter that gets away and maintain ionic balance so that these neurons can function properly. And notice also, oops, I'll start here. Our axon has myelin on it, hundreds of layers of phospholipids wrapped around the axon in all these places. Now, this is done in the peripheral nervous system. By cells known as Swan cells. These are similar to the oligodendrocytes. In that they form and like to form myelin sheath. However, if you remember, the oligodendrocyte can wrap around many pieces of axon, whereas the Swan cells wrap their entire body around just one small piece of axon. So notice this right here is a single Swan cell. This right here is a single Swan cell. This right here is a single Swan cell, and so on and so forth. Another single Swan cell, and again, another single Swan cell. Here, we actually see the process by which this works. Notice the Swan cell grabs onto a piece of the axon and wraps itself around it hundreds of times. And that's really the key to this myelin sheath. Myelin is formed by hundreds of layers of phospholipids wrapped around a piece of axon. All right. With me so far? Now, there's two other implications to this. Notice it doesn't completely cover the axon. There are small gaps in between the Swan cells. These small gaps are known as the nodes. And you can see that right here, these spaces in between. Of course, these nodes were discovered by good old Bob Ranvier. So you may also sometimes hear them referred to as the nodes of Ranvier, which again is incredibly fun to say. Love saying nodes of Ranvier. But you may just use the term node. And it's French, so of course it's spelled nothing like it sounds. But the second point that I want to mention for you is while the Swan cells produce our myelin, they also help the unmyelinated axons. Notice here we see some examples of some unmyelinated axons. Remember, our unmyelinated axons are like the copper wires in our wall, and we don't want our signals crossing. So with unmyelinated axons, we still need to keep them separate. We have to keep the axons separate. So if you notice here, our Swan cell grabs a handful of axons and pushes them away from each other. But notice when it does that, it's not wrapping around them. So it's not forming myelin. It's just merely holding them away from each other. So these would be examples of unmyelinated axons that still need to be separated. We don't want those wires crossing. But most Swan cells, because most axons are myelinated in the peripheral nervous system, will wrap around each individual small pieces. I think I have another nice picture that shows this. I guess I got to clear that. Here we do. Okay, excellent. So again, our myelin sheath is many, many hundreds of layers of our lipids covering and surrounding, wrapping hundreds of times around the axon. Whereas here, notice our Swan cell is just keeping unmyelinated axons from touching. Here, we see an up-close view of two Swan cells, one forming a chunk of myelin on this part of the axon, one forming a chunk of myelin on this part of the axon. And as we mentioned, there is a little bit of a gap in between, known as the node, which, and again, neurofibril node, you don't have to use that. Just the term node is fine. But like I said, if you want to be fun, node of Ranvier, like I said, is very, very fun to say. And of course, remember the two cells, that produce it are in the central nervous system are oligodendrocytes. And in our peripheral nervous system, it's the Swan cells. Insulation. But remember, the most important function of this myelin is that it speeds up the action potential. All right. Excellent. And there, we are now done with the anatomy of our neuroglial cells and their functions. Questions on that? All right, the last thing we need to do from a lecture standpoint is we need to start talking a little bit about how our neurons function, how they produce an electrical signal. I warn you that this part of the lecture is probably the part that is most dense. And I know it's rough because it is at the end of the lecture. So even though it's a little bit early, I want to take one last break before we go into this, because like I said, this I think is the most densest stuff. The good news is after we get through all the dense stuff, we will finish off with the take-home message, the important part of this that you absolutely positively need to know to be successful moving forward on this. But we need to build to it. We need to set a little groundwork first. So we need to talk about that groundwork. We need to be able to put these things together and make sure we make sense of it. And that's going to take a little time, a little effort, a little work. So let's go ahead and take one last break. We will restart at 11:15, and I will start the recording at that point. So the good news is we only have one more part to go. And then, like I said, based on the time, we'll just do the briefest introduction to the anatomy stuff we need to worry about. But I do want to take a little bit of a break first before we do this to make sure that it makes sense, because like I said, it does get a little tricky. This is the denser stuff. So let's take a 15-minute break, come back, and we will kill this off. All righty. Final stretch for the day. So as we talked about earlier, we know that neurons are irritable and electrically excitable. And so as a result of that, we know that when stimulated, they're going to respond by producing an electrical signal. And that electrical signal is called a potential. However, we've thrown the term action potential out there so far, but as we now know, cell bodies and dendrites can't produce action potentials. But that doesn't mean that they don't respond to an electrical signal. Yes, we have an action potential. We know that is a big positive signal, and as we've talked about before, it is something on the order of a 100 millivolt change. It is also all or nothing. What do you think we mean by that?
There's
Like
either going
an
to
on
be
and off switch.
True. Yeah, it's kind of like an on and off switch, and that's a great way of thinking of it, right? We have an on and off switch on the wall for my light here. If I flip it down, it goes off. If I flip it up, it goes on. If I push the lever twice as hard, do I get double the amount of light? Or if I only push it really, really slightly, do I only get half of the light as a result of that? No, it is all or nothing. It's either on or it's off. And it's standard that way. There's no in between. You can't get half of an action potential or 2/3 of an action potential or 1.5 times an action potential or something like that. It is very, very standard, on or off, all or nothing. And as we talked about, it occurs only on the axons. But our cell body and our dendrites also are capable of producing a potential, producing an electrical signal, what we call a graded potential. And again, I like the light switch analogy that you guys mentioned, right? This light switch I can turn on and off in my bedroom, and it comes on and it goes off. However, in my dining room, I have a different type of light switch. I have a dial switch where I can change the intensity of the light, right? If it's just my wife and I together, we want a nice romantic dinner, I turn the light up nice and bright so we can see each other, enjoy each other's company, talk. When the kids are there, I turn it down as low as I can so I don't really have to look at them. They can't look at me. I don't have to see the mess that they're making. I have that ability to change the amount of light. And that's the key with these graded potentials. They are much, much smaller. We're talking about a plus, or minus of around 2 millivolts compared to the 100 that we were talking about as well. But again, it's variable. It can be bigger or smaller. And here's the other big difference as well. While our action potential is a big positive signal, our graded potentials can actually be either positive or they can be negative. So I can have a +2 or a +5 or a +10, but I also could have a -2 millivolts or a -6 millivolts or a -18 millivolts. So we have this modifiable type of graded potential. And as we also mentioned, these graded potentials, I don't know where I can put this because it's going to be in the way here too in a minute. Again, I guess I'll make it small. Nope, I can't do that anymore. Never mind. Cram it in there. There we go. These graded potentials occur along the dendrite and the cell body. All right. Now, we know these are electrical signals, and when we compare it to, again, I keep talking about electricity moving like water, or like the action potentials and graded potentials of our neurons being like the electricity in our wall. When I flip the light switch, the reason my light goes on is because an electron moves across that wire at essentially the speed of light. It is the movement of a charge. That is what it is. And it's the same thing is true for these potentials. These potentials are a movement of a charged particle. And this movement of a charged particle basically produces a current. To produce a current, you basically need two things. You need channels that will allow ions to move into or out of the cell. And you need a starting point. And that starting point is the resting membrane potential. All right. Again, not too much new information. We're talking about graded potential, but we know a lot of information about this. We know that a neuron, when it is at rest, is polarized, meaning that it has a resting membrane potential. And what is that resting membrane potential again?
-70 millivolts.
So it is more negative on the inside than it is on the outside. This is due to a couple of characteristics. We know there's an unequal distribution of ions across the membrane. So let's go ahead back to our drawing board and draw some of these things that we know. We know we have a cell. As you guys mentioned, we know we have a resting membrane potential, which I'll go ahead and abbreviate here, which is -70 millivolts. And as we know, we have different ions that are unequally distributed inside and outside the cell. Like what? Can someone remind me what ions we were talking about that were like that?
Potassium.
Excellent. Calcium. Where is there more calcium, inside the cell or outside the cell?
Outside.
Excellent. A lot more calcium outside the cell. Where's another one?
Potassium.
Where is there more potassium?
Inside.
Inside. Excellent. What else?
Sodium
Where is there more sodium?
outside.
And is it all cations all the time?
Negative.
We've got our most favorite anion, and what's that one?
Chloride.
There we go. Excellent. So we know there's an unequal distribution, large amounts of it in these locations, and let's go ahead and demonstrate that visually by having much smaller letters in the opposite locations. And as we know, we talked about there are both electrical and chemical driving forces on our ions that determine which way they want to go. Which way does sodium want to go?
In.
Which way does calcium want to go?
In.
Which way does chloride want to go?
In.
And which way does potassium want to go?
Oh.
Excellent. All right. So we know that there's these electrical and chemical driving forces determine the way the ions want to flow. Excellent. That's a good starting point. And remember, the other thing that helps us to maintain our membrane potential is the sodium-potassium pump. Right? That sodium-potassium ATPase is a pump, which of course means that it directly uses ATP. And it directly uses that ATP to do what?
Move 3 calcium out and
Calcium?
bring. All right.
What does it move out?
The sodium.
Sodium out. Excellent.
3 sodiums
sodium.
out and 2 potassium in.
Perfect. 3 sodium get moved out. And 2 potassium get moved in. Notice we're having to use ATP because, again, we're moving these things against their concentration gradients. But notice also 3 positive things are leaving and only 2 positive things are going in. So again, that's another one of the factors that helps to make the inside of the cell negative. So we have this unequal distribution of ions, we have our sodium-potassium pumps, and those things help to maintain that resting membrane potential of negative 70 millivolts. And here we see that pretty picture. We have a large amount of ions on the outside, a large amount of potassium, for instance, on the inside. Potassium has the ability to get out, and so we have to pump it back in. Sodium, oh, they have our arrow pointing the wrong way, sneaks in, and we have to pump it back out. So that pump, remember, we talked about in a cell that is at rest, 25% of the ATP is just being used by this pump to maintain the resting membrane potential of the cell. And here's the other thing to remember as well. Our resting membrane potential is equilibrium for the cell. This is where the cell is happy. However, the other important thing to remember, does that mean that there are no ions moving when the cell is at its resting membrane potential?
Cells are always moving.
No, so they're always moving. So the key to this is that there is no net change. For every sodium that comes in, right, two positive, or every positive thing that comes in, a positive thing leaves. For every negative thing that comes in, a positive thing comes in. There's lots of movement. But there is no net change. And again, the key to this is that at the resting membrane potential, the cell is happy. Are sodium and potassium and chloride happy at the resting membrane potential?
No.
No. Ions are 2-year-olds. And what do 2-year-olds care about?
Themselves.
About themselves.
Exactly. All a 2-year-old cares about is themselves. They don't care about you or mom or dad or the dog or anybody else. All they care about is them and what they want. And that's the situation of our ions. Ions care about themselves and only themselves. Like the cell, ions want to be at equilibrium. Right? And again, just like we talked about glucose. We put glucose in the beaker, we poked the holes in the membrane, and glucose moved to equilibrium. Again, equilibrium was where there was no net change. And when we're talking about something like glucose, that's just equal amounts on both sides. However, here with ions, we have to deal with both the chemical and the chemical and electrical driving forces. Not just concentration gradient. So where an ion is at equilibrium, when the chemical force and electrical force are equal and opposite. All right, let's take sodium, for instance. There is a massive amount of sodium on the inside of the cell. So from a chemical standpoint, there is a huge driving force of calcium wanting to come in. Sodium wanting to come in. So if we're going to make the electrical force equal and opposite to that, we need to have some huge electrical force pushing out, where the two would be equal and opposite. And to do that, we need to make the cell very positive. Because obviously, positive things don't like other positive things. So we would have to make it very, very positive. And at that point, these two forces would be equal and opposite. And would it be possible to calculate what that value would be?
Yes.
Absolutely. And so as it turns out, that point where they're equal and opposite is called the equilibrium potential. And that equilibrium potential for sodium, for instance, is positive 66 millivolts. If sodium can get the cell to positive 66 millivolts, sodium will be happy. Again, would the cell be happy at that point? No, but sodium would be happy, and that's all that it cares about. So notice when we open a sodium channel, which way does sodium flow?
In.
Yeah, and we already knew that. When you open a sodium, sodium is going to flow in, and now we know why. Because it wants to make the cell more positive, because it wants to get the cell to positive 66 millivolts. Potassium also has an equilibrium potential, where its two forces are equal and opposite. And in the case of potassium, the equilibrium potential of potassium is equal to negative 90 millivolts. Again, that isn't a piece of information you may have been aware of, but you already knew that because you already know when you open up a potassium channel, which way does potassium flow?
Out.
Out, because that negative ion leaving the cell, the positive ion leaving the cell makes the cell more negative. And it is trying to get it to its equilibrium potential. So think of it this way. When the cell is at rest, who's happier, sodium or potassium?
Sodium,
Well, who's closer to their equilibrium potential when a cell is at rest?
potassium.
Potassium, right? So notice one of the things that we've talked about is we've said numerous times that sodium really wants to get into the cell. Remember, we talked about how we can let it into the cell to even get it to do work for us by moving other ions with those code transporters. Now you can see why. Is because the cell is so far away from where sodium wants to be. Potassium's pretty close. So he's not going to feel very strongly about wanting to move. Yeah, he'll move because he wants to be at equilibrium, but he's already pretty close. So he's not going to have the driving force that's sodium. Sodium is way far away from its equilibrium potential. And so it really, really wants to get inside. And we knew that. So again, there really isn't a lot of new information right here. I know it seems a little dense. I know it can be a little confusing, but there really isn't any new information. And most importantly, here is the big take-home message. The big take-home message is that ions want to be at equilibrium. All an ion cares about, all sodium cares about is sodium. It doesn't care about potassium or chloride or calcium or the cell or anything else. And ions want to be at their equilibrium potential. So if we know their equilibrium potential, we know which way they will flow when we open a channel. And we know that if we open up a sodium channel, sodium's going to come in. We know if we open a potassium channel, potassium is going to flow out. All right? And those are things we already knew. Now we understand why. Because they want to get to these equilibrium potentials. And notice, as I've said many times in this class, numbers aren't hugely important. These numbers, along with this one, and there's probably one other number we've talked about that is really important as well. Any idea what that other? I'll do this in pink down here to the side because it isn't really relevant to what we're talking about this second, but it will in a second. What's the other number we've talked about that seems like it might be pretty important?
Negative 60.
Yeah, negative 60 millivolts. What was that negative 60 millivolts again?
Threshold voltage.
Yeah, threshold to open voltage gated channels. These four numbers are absolutely positively important numbers that we want to know and need to know and are required to know. So you're absolutely correct. Those are important numbers. Equilibrium potentials, resting membrane potential, threshold potential. These are all important numbers. All right. So notice, oh, go ahead.
No, I was going to ask if the electrical or the chemical is more driving force or stronger, I guess.
Great question. Obviously, it depends on the membrane potential, right? However, that being said, when a cell is at rest, for most of the important ions that we're talking about, let's say it this way, the driving force for most ions is more determined by the chemical gradient at rest. Obviously, when it's not at rest and doing work, that can change. And because it can change, that's what makes this equilibrium potential so important. Because we don't really care which one is the greater driving force. We just know that whatever we do to the cell, the ions are going to move to get to their equilibrium potential. Let's say I had a neuron plugged into a machine and I made the cell positive 100 millivolts, and then I opened a sodium channel. Which way would sodium move in that case?
Out.
Yeah, it'd move out because it wants to bring it down to positive 65. So notice we don't have to worry whether the chemical or the electrical force is greater at that point. We just need to know that it always wants to get to equilibrium. So if I give you any value for the cell, if you know the equilibrium potential of sodium and potassium, you know which way they're going to flow. And that's the nice thing about these numbers. We don't have to worry about what the relative values of these two things are, because we know at this equilibrium potential, they're going to be equal. Okay. Great question, though. I like that. All right. Excellent. So
I guess technically, wouldn't the electrical be more of a driving force?
yes,
Because
you are correct. At that point, electrical would be more because it was forcing the sodium out more than sodium would be coming in. So yeah, we can figure it out. But notice it isn't important to take that step. We just need to know which way it's going to go because we'll know which way it wants to go because we always wants to get. If it's above it, it's going to bring it down. If it's below it, it's going to bring it up. We know what it wants to get to. But yes, technically you're right. The electrical would be greater at that point. Now, I'll also tell you the cell never gets to positive 100 millivolts on the inside. Anyway, so again, but if we had an artificial neuron that we could manipulate however we want, we could have that occur. Excellent. So, as we talked about, the chemical and electrical gradient determine our equilibrium potential. And again, we did that. We figured out the equilibrium potentials. We understand those driving forces.
could you go back to that one?
My picture?
Yeah, that wasn't in the slides.
What wasn't?
Oh, the one you were just at.
Oh, sorry.
Yeah.
Alrighty. So, yeah, our equilibrium potential, right, is going to be determined by the chemical gradients and the electrical gradients. But remember, it doesn't matter how strong the driving force is unless the ion can get into or out of the cell. So obviously, having channels is going to be vitally important for this process. And luckily, we've learned pretty much everything we need to know about channels, almost everything. We've talked about all of these. We may not have talked about them this way, but we have definitely talked about them. There are two main types or classes of channels. The first one are what we call leak or gated channels, right? This is like if you took the door off your door frame, or this would be like the archway from your living room to your kitchen, right? Most houses, there isn't a door you can close between your living room and your kitchen, right? They're pretty much open to each other and things can constantly move back and forth without any way of stopping it. This is one of the reasons why we need that pump, because our cells have sodium channels. Sodium's constantly coming in. Our cells have potassium leak channels. Potassium is constantly leaking out. So that's why we have to constantly be pumping them, getting them back where we want them to go. However, if they're open all the time, can we modify them?
Yeah.
Well, if they're always open, then how do we modify them?
It's the Sims.
I don't know what you're going for, but I'll tell you the quick answer. No. If they are open all the time, yes, okay, maybe they're toxins and stuff that we could do it. We could do it artificially that way. But in a normal, healthy, functioning cell, these are always open, so they can't be modified.
I'll say the Sim porter and the antiporter.
Okay, but those are not channels. Those are not leak channels. Those are transporters, right, that are using ATP. These can't be modified. And that's fine, but if they can't be modified, can we use them to do work?
Yes.
They can't be modified, and we can't change the amount of ions that are coming into or out of them. Can we change from a resting state to an active state if we can't change the number of ions that are going in and out of them?
No.
No. So we can't use them to do work. They're just our standing point. Like, again, in your house, even with all the doors closed, is air still able to get into and out of your house? Yeah, there are cracks on the door, there are cracks on the window, right? There's other ways that the air can get in and out. And you can't really use that air coming in and out to do any work. However, you can turn a fan on that can force air to move around, and that does work for us. We are able to do things that way. So these are there, they're present, we're aware of them, but they're not very useful. To do work, we have to produce a current. We need to be able to control, or more specifically, let's do that, change the flow of ions into and out of a cell. And to change the flow of ions, luckily we have gated channels. How many different types of gated channels have we actually talked about in this class?
Like 4 or 5.
I don't think we've gotten that. Okay, you're right in there. There's different types. But if you think of the different types of keys that can unlock channels, what were the kinds of keys that we saw?
Voltage gated.
Voltage and
Chemical.
chemically gated. Absolutely. Well, it turns out, in fact, we've actually talked about 3. The 3 types of gated channels are voltage, chemical, and mechanically gated. Remember, as we talked about, voltage gated is where we change the membrane potential. This is going to be too big. And we have to change it to a critical point. What was that critical point?
Negative
-60.
True. And what did we call that? Negative 60?
threshold.
Threshold. Excellent. Chemically gated, obviously, is where some molecule is the key that opens the channel. Acetylcholine is the most common one in the peripheral nervous system that we've talked about. And even though we didn't use the term mechanically gated channel at the time, remember when we were in the skin, we talked about those Pacinian or those lamellated corpuscles that had many layers like an onion? And remember, we talked about how when they squeezed them, that stretched out the membranes. And when we stretched out the membranes, that opened channels. Well, the fancy word for that is a mechanically gated channel. And so that's what happens here. A force changes the shape of the membrane. And when we change the shape of the membrane, that either opens or closes channels. Here we have it in words, but let's take a look at this with the pretty pictures, because again, these are things that we've drawn before. Notice here is our chemically gated channel. Notice in this case, without our key in the lock, it is closed. However, when our key comes in and binds to it, in this case, notice our key is acetylcholine. We're familiar with that. When acetylcholine binds to it, suddenly the channel opens and ions can flow. All right, again, no new information. We've talked about these. We saw those acetylcholine gated cation channels on the motor end plate. was a great example of that. We've also talked about voltage-gated channels. Notice with a voltage-gated channel, there is, let's do it this way. When the cell is at rest, there is what they call an activation gate, which is closed. When it reaches a specific membrane potential, what we call threshold, then that causes the gate to open. Now, this is a very simplistic drawing. Your book's got a great illustration that I'll show you in just a minute, but I'm going to draw one for you as well, a little bit of a basic one. But what we have here is our plasma membrane. And embedded within that plasma membrane are a bunch of proteins that form our pore. They form the channel. Now, what's special about a voltage-gated channel is they have, as I mentioned, this activation gate. The activation gate is actually usually made up of two proteins. And these two proteins tend to be very positively charged. So what happens is when we depolarize the cell to that magical point that is threshold, what actually ends up happening is that activation gates, or the activation proteins, swing up out of the way. So what'll happen is, and I can't do this with the drawing, so I'll just, nope, hold on. Basically, this swings up and out of the way. Like I said, your book has a great picture that shows this in a minute. And when they swing up and out of the way, then our channel is open and our ions can flow. So channel, channel. And we'll just kind of draw those, pretend those are the activation gates so that they're up and the out of the way so that ions can flow in or out. All right, you with me so far?
You.
Excellent. And again, if you think about it, other than explaining how these doors open, there's really no new information that I've put here. However, there is a bit of information we do need to add. It turns out there is a second gate on some, not all, but on some voltage-gated channels. It is formed by two proteins. One of these proteins is a long linear protein, and the other protein is a large globular protein. And these proteins have incredibly technical names to them. They are referred to, again, they are the inactivation gate, but they are also referred to as the ball and chain. Again, very, very technical terms. Who knows how they come up with the names for these things? What's interesting about this is here, clearly we have a channel that is closed. Ions cannot flow through them. Here, we have a channel that is open. Ions can flow through them. But what will happen is that the ball of the ball and chain can actually wedge itself into the pore. And when the ball wedges itself into the pore, even though the activation gate is open, can ions flow through this channel? No. And so when the ions cannot flow, this special state, this third state, for some of these, we call this channel inactive. So once a channel is inactive, ions can no longer flow. And the only way we can get ions to flow is to get that ball out of there. And it turns out the only way to get the ball out of there is to close the activation gates. When the activation gates come back down, they pop the ball out of the channel. So notice what that means. The gate started closed. From closed, it went to open. And from open, it went to inactive. Once inactive, it must close again before it can open again. It is not possible to go from inactive to open. This is not possible. It has to close first, then it can open, and then it'll inactivate again, and then it has to go back to closed. So the only way to get that ball out of there is to close the gate. Once we close the gate, it pops out the ball, and then we can use this channel again. So basically, once inactive, we cannot use the channel again until it closes. Okay? That is the only new piece of information we have added to this. And if we go back to our really simple example, notice here, they show us this. Notice here, our activation gate is closed. Our inactivation gate is open. So in this case, our channel is closed. Here, both gates are open. Our channel is open. But notice here that inactivation gate, that ball has wedged itself in there. Even though the activation gate is open, ions can't flow, and our channel is inactive. And again, once inactive, it needs to close before it can open again. So again, take-home message is for some voltage-gated channels, there are three states: open, closed, and inactive. And here's that third type that, again, we talked about, we just didn't think of it in these terms. Here we have our Pacinian corpuscle at rest when it is closed. When we then squeeze the skin, that pressure pushes on the plasma membrane and opens the channel. And when it opens the channel, ions flow in and we feel the pressure. When you stop grabbing the arm, the cell goes back to its original shape and the channel closes. So we have three types of gated channels. Wow, that blue doesn't show up at all. Three types of gated channels. And these gated channels allow us to change how many ions come in. And if we're changing the amount of ions flowing, we can do work. Because affecting the number of ions are going in, again, if you think about it, that electron on the wire is the movement of a charged particle, a movement of an electron. A charged ion entering or leaving a cell basically is the movement of a charged particle and that movement of a charged particle we have a fancy name for in physics. We call it a current. And in neurophysiology, a current is a potential. And I think I say that on the next page. Oh, here's the pretty picture from your textbook. Notice this one does a much nicer job of showing us the anatomy, even better than what I was able to draw. Again, we have the proteins that are making the pore. Here are those positively charged activation gate. When we depolarize it, whoops, to that magical point to threshold, then those gates swing up. And when the gates swing up, the channel is open and the ions can flow. But when the ball of the ball and chain, that inactivation gate, wedges itself in there, ions can't flow anymore, even though the activation gate is open. So it is now inactive. And notice the only way to get that ball out of there is to punch it out by closing the activation gate. So that's the other key take-home message. To use a channel again, we must close the activation gate to remove the block. And then once we remove the block, then the channel can be opened again. All right. So now we know how to change the number of ions that flow. Thanks to our equilibrium potential, we know which way the ions are going to flow. And what was the equilibrium potential of sodium again?
-66.
Negative 66?
Oh, positive 66.
Excellent. What was the equilibrium potential of potassium?
-90.
There you go. So we know which way ions are going to flow. We know how to open channels to get those ions to flow. When we get those ions to flow, we produce a current. A charged particle moving into and out of the cell is a current. At least that's what they call it in physics. But here in anatomy and physiology, we call that a potential. So that flow of charged particles into or out of the cell is a potential.
I do have one question, Professor.
Yep.
So if the resting membrane potential is -70 and it gets hit by a +100, wouldn't that take it to +30 and close it immediately?
So say that again.
So the resting membrane potential is -70 millivolts,
Correct.
and it gets hit by an actual potential of +100. Wouldn't that make it +30 and close up immediately?
Close up what?
Inactive, the inactivation.
So you're I love the way you're thinking about this, and you're absolutely correct. However, the one thing that I would say is that it isn't so much that the neuron is being hit by an action potential. The neuron is going to produce an action potential. And guess where it produces that action potential? Wasn't there a go ahead?
Trigger zone.
There you go, the trigger zone of the axon hillock. Absolutely. We are going to stimulate this neuron, and if at the trigger zone we can get a big enough change, then it will produce an action potential and it'll spread. And you're right. Not surprisingly, the way we produce an action potential is a big positive signal, a big, huge influx of sodium. But notice sodium doesn't get to its equilibrium potential. You're right. If it's only 100 millivolt change, we're only going to get to plus 30. And that means that sodium's not getting happy. So something else must be happening. And yeah, one of the things that's going to happen is inactivation. All these things that I'm telling you, while they may seem a little dense, they're not just random. These are exactly the concepts that we're going to be using to understand exactly how an action potential works.
I was thinking more along the voltage gated channels.
But remember, the voltage gated channels are a light switch. When you go above threshold, you turn it on. So if you push up on your light switch, right? If you just touch your light switch, nothing happens. If you push it a little bit, nothing happens. A little bit more, nothing happens. But when you push with enough force, it flips up and the light turns on. If you keep pushing with more and more force, does the light go off? No. Once you pass threshold going up, our voltage gated channels open. The only time the voltage gated channels close is when you go back below threshold. When you go back below threshold is when they're going to close. So it doesn't matter how you go above threshold, the voltage gated channels stay open. But you're right, we do have those pesky inactivation gates to worry about. And that is something we absolutely positively will talk about. But now that we've laid the groundwork, we're going to save that for the next class. In the next class, we are going to talk about our potentials, both graded potentials and action potentials. We're actually going to see how we produce them, what ions are involved, what channels are involved, and what effect they have on the neuron. So first, we're going to produce a graded potential, then we're going to make an action potential, and then we're going to spread our action potential. And we're going to spread that action potential both with and without myelin to see the difference. But looking at the time, that is information for the next class. So we've lit the groundwork. Our goal now is to learn and understand how we make our two types of potentials, both graded and action. All right. So that's it for the lecture for today. Any questions on that? Like I said, and as I knew we were going to do, we did go long on the anatomy. So I'm not going to do too much on the anatomy, but there is one point I wanted to emphasize. And to do that, let's go here. As I mentioned, oops, not that one. I want this. As I mentioned, on this next lab exam, you have that brain anatomy handout, which, as I mentioned, is going to be about 50% of your lab exam. It's a lot of material. Basically, you're responsible for three things: structures of the nervous tissue, actually four things. You're responsible for the structures of the nervous tissue. You're responsible for, of course, our histology, which there's more of on this test than there were on the past two tests. The last thing you're going to be responsible for is, remember, unlike our muscle, here the muscle in my bicep brachia, all of this muscle tissue does the exact same thing. So if I took a little bit of muscle here and I switch places with a little bit of the muscle here, they do the same things. We know that's not true for my nervous tissue, right? This tissue here doesn't do the same thing as this tissue here. Different locations have different functions, right? And that's one of the problems with this one, because it's nice. We drew the origins and insertions of the muscles because it helped us to know what they would do. Does the part of your brain that move your foot look like a foot? No. So there's a little bit more memorization that goes on this. So what we need to learn are the regions, or what we can also call the areas of the nervous tissue, And for those regions and area of the nervous tissue, we need to know their functions. Right? Just like in our muscle system, a structure is a structure. A structure doesn't have a function. It's a nook or a cranny of the brain. Regions have specific functions. And that's what your list is. Your list is a list of structures, regions, and functions. And it's a lot of information. So in the bones, I just didn't give you a random list of bones. We broke it up by parts: axial, appendicular, skull, rib cage, so on and so forth. Muscles, we broke up into more easily digestible parts: muscles that move the head, muscles that move the arm, muscles that move the leg. So we are going to break it into more easily digestible pieces for our nervous tissue as well. And those more easily digestible pieces are what we refer to as the cephalons. Cephalons are divisions of the nervous tissue. And these divisions occur very early in our development of the nervous tissue. Basically, when our nervous tissue starts to form, it forms as a flat sheet, basically what is called a neural plate. That neural plate gets a tiny little kink in it that we call the neural crest. That neural crest allows it to roll up into a neural tube. That neural tube, as you notice, typically starts to develop around week 2 or 3. And by week 6, we have these 5 distinct enlargements to the brain that are known as the cephalons. They are the telencephalon, the diencephalon, the mesencephalon, the metencephalon, and the myelencephalon. I know these are 3 M's, and that gets a little tricky, but if you notice, they're in alphabetical order: mes, then met, then my. And I'm not just randomly picking these cephalons as our dividing points because they're A, hard to spell, and B, hard words to say. But if you notice here, starting at 6 weeks, our nervous tissue has already started to differentiate to what is going to form our distinct regions of the brain. So these 5 bumps in our neural tube grow up to be these 5 regions of our nervous tissue in an adult. And notice they've color coded it, so the beige becomes the beige, the pink becomes the pink, the green becomes the green, the blue becomes the blue, and the, I don't know, whatever that is, reddish orange becomes the reddish orange this here. Notice the telencephalon becomes the structure known as the cerebrum. These 2 terms are basically interchangeable. All of these nooks and crannies that we see in this big structure that looks kind of like a big mitten. I always think of the brain as looking as this big mitten. You have this big, huge, lumpy thing, and then a thumb sticking off to the side. So this whole big, huge, with all of its nooks and crannies, this structure, what we typically think of as the brain, is your telencephalon. Deep at the center of the telencephalon, underneath it, is a structure that is formed from the diencephalon, and if you'll notice, is still called the diencephalon. The region that is the mesencephalon is still called the mesencephalon, although I will warn you, it is sometimes referred to as the midbrain, so you will see that name for it as well. Notice the blue part becomes these 2 big enlargements, the pons, which is an enlargement to the front, and the cerebellum, which is an enlargement to the back, which, if you remember, we mentioned earlier, is where you're going to find those Purkinje cells here in that cerebellum and the back inferior part of the brain. And the lowest part of the brainstem, the myencephalon, becomes your medulla oblongata. So what you'll notice on your list is all of the structures, all of the regions, all that you are responsible for, I've divided into these 5 different sections to help us to make the information more easily digestible. Now, of course, it makes it more easily digestible, but these are terms you're responsible for on the exam. So on the exam, with a picture like this, could I have an arrow pointing here? And could I ask you to identify this cephalon? Yeah, absolutely. And if I did that, what would your answer be?
Cerebrum.
Now, what cephalon is it?
Oh, telencephalon.
Yeah. Or I could have this arrow and instead have it point to this part down here and ask you to identify the cephalon. And what would your answer to that be?
Myelincephalon.
Yeah, myelincephalon, exactly. So that cephalon is a question that I could ask on the exam, but it's also a way to more easily digest this information. Like I said, we will go through these lists together. But as I've also pointed out, your exam is for this materials on the 29th. What is today? Today's the 16th. You basically have 2 weeks from yesterday to learn this nervous stuff. So going into the weekend, even though we will go over the brain anatomy together, remember, like we've done in the past, it'll be quickly, and I don't expect you to have mastered the information. So I strongly encourage you to spend some time this weekend to start working on the list. It's okay if you can't answer all of it. Figure out what all of it is, that's okay. But even if you're exposing yourself to it and trying to figure some of it out, it will help tremendously when we're going over it together. All right.
So which handout are you talking about specifically?
Specifically, I am talking about under your lab handouts. You have your histology, which again, the histology is part of it as well. So I would say these 2 things together, the histology and the nervous system anatomy, these things combined are going to be 50% of your lab exam. But specifically, the handout that I'm talking about right now is this one, central nervous system anatomy. So when you look at this handout, notice here, it reminds you of the organization of the cephalons. Here are the telencephalic features. Notice many of these things, cerebrum, cerebral cortex, gyrus, sulcus, a lot of these things are just structures. So these first things are just structures. So really, all you have to do is be able to identify these on a model or a chart or a brain or something like that. These are the structures. However, notice down here in the lobes are some specific areas. And let's start easy. Broca's area happens to be an area. You need to be able to find that area on a brain, but you also need to know the function of that area. And guess what the area of Broca's area, which is also known as the speech area, is?
Speech.
Yeah, it's where you produce speech. Guess what the function of the primary visual area is?
Vision.
Yeah, see light, exactly. So for these areas, you need to know their functions, right? A gyrus doesn't have a function. A sulcus doesn't have a function. They're just structures that need to be recognized. So this list, along with the histology, is going to be 50% of your lab exam. So start trying to identify, see if you can find the structures on the pictures, on the charts. And I've got some unlabeled pictures and charts from your textbook that are there. There are other great resources. And then also for these, find the areas. And for the areas, you also need to know their functions. All right. And again, that's in the lab handouts. All righty. Well, I don't know about you, but I'm exhausted. Questions on any of that? All right, excellent. Then you guys have a good day. Have a good weekend. Study hard this weekend. You only have 2 weekends to study for this. Well, two-day weekends. You also have 2 one-day weekends to study for this exam, but you only have 2 more 2-day weekends to study for this exam, and this is one of them. So please take advantage of it. I know we just introduced this stuff, but hopefully we've given you enough to keep you very, very busy this weekend. All right, any other questions?
Just to clarify, like the final test schedule.
Yeah.
It looks like our last lab and lecture exam is, I think it was Wednesday, and then the final is just that following Thursday, correct?
Close. Remember, we're not in class on Wednesday. So Thursday is your lab and lecture exam. So again, just like yesterday was your lab and lecture exam, solely focusing on the nervous system. So you will have a lab exam solely on the nervous system, a lecture exam solely on the nervous system. And then you'll have a whole 24 hours to prepare for your cumulative final exam. Now, the cumulative final exam, and again, this is, I'm glad you asked this question because I don't think we've talked about it yet. It is going to be 100 multiple choice questions. So there are no essays on the final exam. You are just going to be responsible for all of the material, but just recognizing the correct information. So the key to that is be able to recognize information. You don't necessarily pull the information out of the ether. However, that doesn't always necessarily make it easier. Remember, almost by definition, multiple choice questions are tricky. They've got that one word where if you misinterpret that word or you misread the question, they're going to have that trap answer to catch you. So you have to read the question so you can answer them correctly. The other thing, I saw Brian's enthusiasm about not having essay questions, but remember, there's one other very magical thing about essay questions. Essay questions give you partial credit. Yes, if you know all the information, you get all the points. But even if you know some of the information, you get some of the points. That's not true in a multiple choice question. You could totally own a concept. Hell, your uncle could have been the one who discovered it. You could have had your picture taken with that concept, know it like the back of your hand. But if you misread one word, you misinterpret the question. They have that trap answer. And even though you totally know that piece of information, you're not getting any points on it. So again, while I know there's enthusiasm that it's going to be multiple choice questions, make sure you read the questions carefully so you can answer them correctly. And it is going to encompass everything in the class. We've been basically broken out into 4 sections. So each section will have somewhere around 25 questions involving it. Some add a little bit more, some add a little bit less, but again, it'll encompass everything we've talked about so far in this class and still have yet to do. All righty. Did that answer your question?
Yeah, thank you.
Excellent. All right, great question. Any others? All right, excellent. Well, then have a good weekend, study hard, take care of yourself. Apparently Delta's going crazy. More and more they're encouraging people to start wearing masks again. More and more people are having breakthrough infections, although the breakthrough infections are typically less significant than the others. So if you haven't been vaccinated, I strongly encourage you to get vaccinated and be safe, be smart, listen to the scientists, not the politicians. And I will see you guys on Monday.
Thank you.
All right, bye.
Tag.